All right, so before we get started, I just want to go through the BioData Catalyst standard statement of conduct. The Catalyst Consortium is dedicated to providing a harassment-free experience for everybody, regardless of gender identity and expression, age, sexual orientation, disability, physical appearance, body size, race, or religion, or lack thereof. We do not tolerate harassment of community members in any forum. Sexual language and imagery is generally not appropriate for any venue, including meetings, presentations, or discussions. This statement is also available online, and it is our sort of guidepost which we hold the consortium to and everybody engaging with us. All right, so here's a brief look at our agenda for today. The slides are located in the forum. If you wish to download them and follow them along on your computer, you're welcome to do that. For convenience, the links in the agenda of the slide go directly to that respective section of the PowerPoints. We have a lot of other links to materials and resources throughout the slide, so you may find it beneficial to download the slides for future reference and follow those links. All right, introductions and housekeeping. Without further ado, I'll do some introductions to the BioData Catalyst staff here today, and we'll go through a few housekeeping items. Okay, so we have 4 hosts today. I'm Becky Boyles. This was a picture of me pre-lockdown. My hair is no longer clearly that short. But yes, so I am co-PI of the BioData Catalyst Coordinating Center in partnership with Stan Ahalt at UNC Rensselaer. I am head of the Director of Data Modernization at RTI International. I'm very pleased to be here with a number of my collaborators. These folks will all be doing some part of the workshop today, and they're sitting here in the front. So if you guys have questions, raise your hand. We'll all be working as a team to kind of get through this afternoon. I also want to give you a big thank you to our guest researcher who's taken time to join us today, Ravi Mathers, also in the front. Ravi is a statistical geneticist at RTI's Genomics, Bioinformatics, and Translational Research Center. We finally call that group GoBot. We got pretty creative with the acronym, I think. And he works in novel method development with omic technologies, building fair data portals and integrated data and knowledge across disparate sources. He was a BioData Catalyst Fellow, and his fellow's work, which he'll speak to you about today, focused on matching public controls for new GWAS with case-only controls and building a protocol called, how do you say this, Ravi? GAW-merge, G-A-W merge, which integrates array and whole genome sequencing data. All right, we will save some time at the end of each section for questions, so don't be shy. We will also periodically pause to see if anybody has questions or needs assistance during live demos. We will be using a microphone that we can pass along if you have a question. Because we do have some folks remote on Zoom, we ask that if you have a question or comment that we use the microphone so everybody can hear. If you've joined the community, which are part of the pre-workshop checklist, you will have access to the community forum. There we've created a space exclusively for this workshop. We have remote reps who are listening in to the workshop and actively monitoring the forum for questions. So please feel free to use the forum all afternoon today, anything you want to ask. The space will exist after the workshop ends, so feel free to post questions or engage with others even after this afternoon. Note that we've added today's slides to the welcome post in the forum. So if you want to follow along with us, you can download a PDF there and follow along. All right, any questions before we begin? Okay, excellent, thank you. All right, so I'm going to give a very brief overview of what BioData Catalyst is, and then my colleagues will come up and show you different parts of the functionality of the system during the afternoon today. So we'll briefly talk about where to find more information about the ecosystem, including website and documentation resources. And then lastly, we'll go over why and how you can get involved in the community. So BioData Catalyst is both an ecosystem for analysis, as well as a community of researchers and informaticians who kind of collaboratively engage.
Yeah, now you can do that.
Oh, all right. Thank you, Elisa. All right, so first, we'll start with an introduction. We'll talk about how data in recent years has been growing, how this led to the creation of BioData Catalyst to serve that data, and how we aim to facilitate research through this software application. So let's start by talking about the motivation that led to BioData Catalyst. I assume because you guys are here in this room that I'm preaching to the choir, but we are in the age of big data. The rate of data generated is accelerating extremely quickly, and medical fields are keeping right up with everyone else. Actually, more biomedical data will be generated this year than all previous years combined. And there are many diverse data modalities collecting more and more different types of data, health data, surveys, imaging, et cetera. As you see from the chart, we've taken years for data generation, now really just takes a few days. And that's great, but researchers really need proper resources for storing and analyzing this data. We need a way to do something with the data. We call this within BioData Catalyst sort of democratizing data. What we really want to do is tap into that power to enable research, right? Data for data's sake is not what we're about. We really want to enable that reuse. So even more specifically, we often refer to this slide that shows top med data growth accelerating very quickly. You can see the number of samples and file sizes have grown tremendously in the last few years. We'll be talking more about data access and the amount of data and data sets available in BioData Catalyst later in the workshop. And as I said on the previous slide, data is great, right? But the explosion of clinical and research data brings challenges for researchers using that, and we will delve into some of those today. All right, so today, really, the cloud is a tool that enables us to make better use of the data that's being generated out of these studies. Looking at the graphic on the right, we're really shifting from an old model where the users with their own data sets, usually small data sets, not always, would send you to complete your analysis, and then send back the results. If we want to share data, you had to send it. So a lot of this, gosh, back when I was doing lab work was certainly all in email, but now we have a central computer structure, not only for data sets, tools, and workflows as well. People can come in to the cloud environment within BioData Catalyst and do their work. There are many advantages for this new model. First, it scales, right? I think many of us know the sort of on-prem solutions, and I'm definitely back in the day where I remember sending data and analysis via email and sometimes via hard drives. And that definitely doesn't scale. Secondly, it levels the playing fields. Not all institutions have big budgets for sophisticated compute infrastructure. By providing a cloud platform like this, somebody can come and access elite compute regardless of kind of where their institution sits and what they have access to institutionally. Third, because it's cloud-based, researchers are able to access data without needing to physically copy it. And we can be sure we're all referring to the same data sets and also tools and workflows and not get out of sync. Last, we have the data and methods in a single place, streamlining reproducibility. So if you have a certain approach for a problem, everyone can access that approach and see the results. You don't have to recreate it. So the explosion of clinical and research data in recent years has led to the need for the creation of cloud storage and analysis platforms. Data use agreements limit the ability to analyze data in some cases. Moving data is hard, it's time consuming, it's expensive. We needed to have an infrastructure that allows researchers to bring tools and workflows to the data and not the other way around. And this was really the impetus behind the creation of BioData Catalyst. And NHBLI initiated this effort 3-4 years ago to address the need for HLBS research. The mission is to develop and integrate advanced cyber infrastructure, leading edge tools, and fair data to support the NHLBI research community. The vision is to be community-driven ecosystem. So it's really critical to us that we engage actively with you all. We want to understand your needs. We want to understand what you're doing. It really does drive the future roadmap for the ecosystem. Within BioData Catalyst, our development efforts are purposely user-centric. This keeps us focused on driving scientific outcomes, which are aimed at ultimately improving the lives of patients. I mean, that's why we do this work. Additionally, we work in close partnership with dbGaP and other data repositories to facilitate data access according to their existing processes and to make use of a number of existing data services. We are really engaged heavily across the NIH community to ensure we are leveraging data resources across that community and that we're all aligned. So now, hopefully, you guys have a much better understanding of kind of what has led to BioData Catalyst, what our mission and vision are. That's having an overview of how kind of this works together. We have different types of users and researchers. I mean, this is just a cartoon example. Bioinformaticians, developers, administrators, clinicians, all working with different types of data in the ecosystem. Genomics, clinical data, imaging data. All this data is available in the cloud-based platform, providing tools and applications of works and workflows in secure workspaces. We're committed to collaborative efforts. For example, we have tooling for workspaces for collaborators to work together, so you can collaborate on the same data sets using same tools. All these features help accelerate efficient research that drives discovery and scientific advancement. This is all, again, you're going to hear a lot of this today, but ultimately in an effort to improve patient outcomes. We are the BioData Catalyst ecosystem, a cloud-based platform providing tools, applications, and workflows in secure workspaces. It's designed to be nimble and responsive to ever-changing conditions. And really what makes us nimble is that engagement with you all. So our developers, our development roadmap is really, and I can't say this often enough, directly informed by what the community requests and needs. Science changes rapidly and is part of our goal to keep up with that pace. Okay, so in its current state, what does BioData Catalyst offer? It offers elastic computing, so you pay for what you use, no matter how large or small. It's not permitted by the system. You have access to. It also offers access to high-value data sets and multiple tooling options. There's an interactive browser for exploring data sets and variables. We have statistical analysis tools, and beyond that, there's a number of other specialized workflows. And because we're a user-centric endeavor, we offer the opportunity to be involved in the community, which we're going to discuss in more detail later this afternoon. We have a lot of interaction between the consortium and our users. You can get inspiration, get help. If you're, we have pages online, or you can engage with us directly if you're writing a grant, formulating an idea, and you'd like some support. These assets and the people involved in the community are really a huge resource that's there for you all to tap into. So there's no need to download and manage multiple large data sets. That's a critical functionality within BioData Catalyst. There's no compute system for you to manage. You don't have to worry about upgrading infrastructure or other hardware. You only have to pay for what you use. We have a dedicated help desk and a huge wealth of documentation resources available to help guide you through using the system. Also, we are here to support you. You're going to hear that today. We have members of the consortium who monitor our help desk, who respond to emails, who help people insert language into grants. All this is really how we engage as partners with you all. So what do we mean when we say the BioData Catalyst ecosystem? You're going to hear this a lot. Technically and architecturally, we're a secure cyber infrastructure of interoperable platforms that works together to enable various research-focused use cases. Within the ecosystem, a user can search and browse available data, access workflows, and use the top meta imputation server. Once a user has identified data of interest and data access has been verified, they may bring data and tools into a secure workspace for analysis. Now we're very briefly going to talk about where to find more information about Catalyst. Our website has a number of resources, including links to documentation, tutorials, learning modules, as well as things such as access and information on various platforms in the ecosystem, information about the data we host, and more. So where do you find all this information? The services page on our website has all you need to connect and learn about the platform and services available in the ecosystem. It will help you through the various components, which include exploring available data, analyzing data in a cloud-based shared workspace, and using community tools on controlled access data sets. This page includes links to launch the various parts of the ecosystem, as well as links for documentation and learning modules on each as well. We have two resources listed at the bottom of this slide. The Learn page of our website is where you can get started on the BioData Catalyst ecosystem with our collection of documentation, videos, FAQs, community forums, tutorials, blog posts, upcoming events, and more information about our partners. We also have documentation hosted on Gitbook that covers a huge array of topics for new users. Also, you can post on the forum or contact the help desk. No questions too big or too small. We really encourage you to reach out, engaging with you. Again, it's a huge part of how we work and how we operate and what makes the ecosystem valuable. Okay, so to recap, you can view the services page to learn and launch all the components within BioData Catalyst. We are an ecosystem, which broadly means we're a system of systems. So we do have underlying components, but the services page is where you can access various pieces of that ecosystem. You can view the Learn page to access a variety of resources, including documentation, videos, FAQs, all the stuff I just mentioned, which other users have found hugely helpful. You can access the documentation library via Gitbook and browse a variety of topics. We also have a YouTube channel, which we encourage you to subscribe to if you find video tutorials and presentations to be helpful. Lastly, let's talk about how you can get involved in the community. As mentioned earlier, the primary goal of the project is to build a data science platform, but at its core, we are a people-centric endeavor. BioData Catalyst is building a community of practice to work collaboratively to solve technical and scientific challenges. This is really about the users and what you all want to do. We're really happy you're joining us today, and I just want to mention that we hold community hours every month. Community hours is an hour-long virtual informational session that splits time between a topic that's featured or a new feature of the ecosystem with time for discussion and Q&A with platform and service representatives. A good session to join is our next session in May, which is going to be BioData Catalyst 101. In that session, we'll be going over what BioData Catalyst is and the best ways to get started. It'll be a really good session to join after completing this workshop. And if you signed up for the community, you will receive emails from us as we get closer to that date. So those emails will include information about how to register and an agenda around what will be covered. So also I encourage you, if there are other folks who couldn't attend today, that BioData Catalyst 101 community hour session is a really great starting place. Additionally, you can find all of our past community hours materials, including notes, slides, and recordings on our community forum. There's a lot of beneficial material there. You can find, especially if you're interested in a particular topic. We also have a few recorded community showcases where researchers present what they're working on and how they use the ecosystem, so you can hear firsthand their experience before getting started on a project. If you viewed our pre-workshop checklist, you will notice we had a step to join our community. Joining the community is truly the actual next step if you want to get started right now. And if you haven't done this already, we encourage you to go ahead and do that. You can do it today. It doesn't take very long, a minute or two. We have a short registration form. Note that you can still join the community even if you do not have an ERA Commons account yet. We'll be talking about authorization and access a little later in the afternoon, but make sure you sign up in order to stay informed of upcoming events such as our monthly community hours. When you join the community, you will have access to our BioData Catalyst help desk, our forums, future updates on new releases and events. And after you've joined the community, assuming you have an ERA Commons account, you can sign up for workspace of your choice, apply for cloud credits. It may take, just so you know, applying for cloud credits can sometimes take a few business days for approval, but we'll go through that later in the workshop. And you can get started exploring data even without cloud credits. You can start accessing and practicing in the workspaces. So just to kind of recap, that was a lot of information I just went over. To get into the system and start working, you do need an ERA Commons account. You do not need an ERA Commons account to join the community. So if that's a barrier for you, you can get started there while you're waiting on your ERA Commons account to be assigned. All right. I feel like that was the most talking I've done in about 2 years. Are there any questions? Okay. All right, we're going to start with an interactive demo on finding and using hosted data from Emily Hughes from our pictured team at Harvard. So while we give Emily a chance to come up, again, feel free to raise your hand or ask questions or put them in the Zoom chat at any time. Thanks, Emily.
Alrighty, can everyone hear me? Is that good? All right. Awesome. Well, thank you, Becky, for that introduction. As she mentioned, my name is Emily Hughes. I am here as part of the Picture platform of BioData Catalyst, representing Harvard Medical School. So I work as a bioinformatics systems analyst for Paul Aviak's lab. All right, so jumping right in, what data is available in BioData Catalyst? So BioData Catalyst hosts a number of different controlled and open data sets, the majority of which are part of the TopMed initiative, so the Transomics for Precision Medicine. And these include many genomic files such as CRAM files and multi-sample VCF files, in addition to phenotypic data and the harmonized phenotypic data. So there was a TopMed initiative where they harmonized a bunch of different studies. So that is also available on BioData Catalyst. Additionally, we have the 1,000 Genomes Project and several COVID-19 data sets. So we have the ORCID and the Red Coral data sets. Additionally, we have teaching data sets, so training data sets from BioLink. These include Framingham and CAMP, and some sickle cell disease specific data sets, including Baby Hug, Walk Fast, and Stop 2. We are also always ingesting new data sets, so some of our coming soon include additional BioLink teaching data sets and clinical trials data sets. We are also hoping to get more sickle cell disease related data sets from the Cure Sickle Cell Initiative, and additional TopMed data, COVID-19 data, and the Pediatric Cardiac Genomics Consortium data, or PCGC. So that's a big list. So I was hoping to show you a bit more visually what sort of data is hosted in BioData Catalyst. So we have about 3.42 petabytes of data on BioData Catalyst, which consists of more than 280,000 participants, 490,000 data files, and 150,000 whole genomes. Additionally, we have data from many different ancestries and ethnicities, as well as different phenotype focus. And I believe our guest researcher will be going over this breakdown a bit more later this afternoon. So that's great that we have all of this data, but how can you check your personal access to the data? On BioData Catalyst, there are 3 main ways to do this. The first is through the BioData Catalyst website that Becky had pointed out earlier. The second is through one of the data exploration platforms that we have called Gen3. So I'll be demoing how to use that platform a little bit. And then the third way is the picture platform, specifically the data access dashboard. So let's go ahead and jump right in. I'm going to see what happens here. One moment while I get my screens figured out. Okay, because it's in presenter mode. It's all right, I'll just. Oh, I see. Okay. Excellent. Okay, perfect. That's okay. Yeah. Awesome. Okay, so this is the BioData Catalyst website. I'll go ahead and go to the home page here. So when you go to biodatacatalyst.nhlbi.nih.gov, this is the home page for the BioData Catalyst website. And so if you go to resources and then data, you should see this check my access button. And so this will actually have you log in with your ERA Commons. I'm already logged in on this computer, so I'm going to click yes, I authorize. And then here we can see Elisa has access to all of these studies here. So it provides you a list of all of the projects that you have access to. So this is one way to check your access to data. Another is through the Gen3 platform. Yeah, I think we're good. So when you navigate to Gen3, this is what the Gen3 BioData Catalyst powered by Gen3 looks like. And this really allows you to get kind of a quick summary of the data you have access to, as well as looking at the file level of the data. And so here you can see we are in the exploration tab here. And you can search either by the data or by file if you're interested in this. So this actually provides you a list of all of the files that you have access to for each study. And over here, you can see we have different filtering. So if I'm curious, maybe in the COPD gene study, I can filter to just that consent group and get the files for just that group. Additionally, there's this discovery tab, which gives you a different kind of overview of the data. And you can filter this by what is available to you or maybe not available. So this kind of allows for a good overview of kind of some study level metadata. Yes, I think we've got a question over here. And please, if you have any questions, raise your hand.
So is this data still from the access still coming from the BioData application? So is that how they know essentially what you get access to?
Exactly. Yep. So because we use the ERA Commons ID, that information is communicated from dbGaP. So these are the studies that you are authorized to access from dbGaP. Yep. All right. Any other questions before I move forward? All right. Great, and so I went through the first two ways to check access to data, and before I go through the third one, I wanted to introduce Picture as a platform a bit more before diving into the third. All right, so what is Picture? BioData Catalyst powered by Picture is a platform that allows researchers to discover data and create cohorts to use for their projects. So Picture stands for Patient Information Commons, Standard Unification of Research Elements. And so this platform integrates clinical and genomic data across BioData Catalyst, including data from TopMed and TopMed related studies, COVID-19 studies, and BioLink data. Picture has a user interface or a UI that allows users to search available data using queries to build cohorts. And once the query is built, these results can be exported via the Application Programming Interface, or API, into an analysis workspace of your choice. And so I also put the link to BioData Catalyst powered by Picture on this slide here, which you can also access from the BioData Catalyst website. So to fully understand the usefulness of this platform, first we need to understand the complexity of the traditional workflow when organizing, searching, and querying data. So this slide, I won't go into the details of this slide, but it provides a brief summary of what this traditional workflow could look like. So as you can see, you start with downloading the data dictionary XML files and the data table text files from your study or data set of interest. And then these files need to be parsed for your variables of interest, and then you need to map it to the data itself, parse again for the raw data to be mapped and decoded, and then finally we can get that data into a usable data frame to start our analysis. So BioData Catalyst powered by Picture simplifies this workflow tremendously so that you can focus on finding your cohort of interest, getting your data, and starting your research. And so users can do this in two ways, but the method is essentially the same, just slightly different how you do it. So you can do this through the user interface or the UI, or the application programming interface or the API. And so first, you build your query of interest, which I'll show you how to do in a moment in the live demo. And then you select the variables you want to use for your research to export into the final data frame so that you can start that research. And so the UI is kind of a user-friendly point-and-click interface, while the API allows researchers that are more comfortable coding. You can use Python or R to accomplish that same thing. All right. Additionally, there are two parts of Picture. There is Picture Open Access and Picture Authorized Access. So as an introduction, open access allows any user with an ERA Commons credential to search any clinical variable available in Picture. So that means that if I'm a researcher and I have an ERA Commons account, but I'm not authorized from dbGaP to access any data, I can still use this open access to search for variables of interest and do some quick power calculations to figure out what I want to request access to. Authorized access, on the other hand, means you need to have access to at least one study to use this. So this means that you must have authorization from dbGaP to access this data, which then you can export into an analysis platform. So in open access, you can search any destigmatized clinical variable. So this just means that we have gone through and removed some of the sensitive information to help anonymize and obfuscate the data to ensure patient confidentiality. But in Picture Authorized Access, you can search all phenotypic and genomic data because you have access to that data set. Additionally, in open access, you only get aggregate counts, so the number of participants that meet your query criteria. Whereas in authorized access, because you have access to it, you can get patient-level data. And so, as I mentioned, a potential use case would be for open access. If I'm a new researcher starting a new project, this is a more exploratory tool to figure out which data sets to request access to, while authorized access is kind of the first step in building your cohort to do your actual research. Any questions so far about the differences between the two? I'll be going through a live demo later, so hopefully it'll clear some things up, but. Right. We love questions. Okay. All right, so the first part of Picture I would like to demo to you is the data access dashboard. And so the data access tab of Picture provides a summary of authorized and open access in case you can't remember that table I just went through. And also a table view of the available studies in BioData Catalyst powered by Picture. So let's go ahead and get here real quick. All right, so if we navigate to the Picture URL, we will log in with our ERA Commons. Elisa is already logged in on her laptop, but you would usually go through the process of logging in. You click Yes, I authorize for Gen 3. And this is the landing page that you end on. And so depending on what you are authorized to access, this page will look different to you. Elisa here has access to all of the studies. So here you can see under authorized access that she has access to the 61 studies and over 280,000 participants. It is the same for open access. So everyone's open access should be the same because you are able to view any of the studies and any of the participants, regardless of your authorization to access data. And again, here's a quick summary of the differences between the two types of access in picture. And if we keep scrolling here, we have our kind of data access dashboard table summary. So this lists all of the studies that are available in picture, as well as some metadata associated with them. So here we have the PHS accession number, the study code, the type of data, the full name and consent information, as well as some additional metadata. So number of clinical variables, number of participants, number of samples sequenced, things like this. Additionally, in this last column over here, we see access. And so because Elisa has access to all of these studies, it says granted for all of them. However, if I didn't have access to one of these studies, I would be able to hit this button that says request. This would actually take you to the dbGaP page for that study, so you can learn some more information about it and submit a formal data access request through dbGaP to kind of get started on that process. All right, that was a lot. Oh, I think we have a question over here. Okay, so I think, let me make sure I understand your question first. Are you saying all studies in BioData Catalyst are on picture? Was that your question? Yes. Yes. Yep.
This is a fabulous one question that was independent in the ability of groups to upload the data.
Yep.
Which is very resource intensive. Is there going to be any additional resources?
Yeah, that's a really interesting question. So just to repeat for the Zoom folks, the question was, this is all dependent on the process of uploading data to dbGaP, which can be very intensive. So is there going to be assistance in uploading data to dbGaP? I am unsure of efforts of that right now. Oh, just kidding. We have an answer.
So we've been working with NIH because we want to do dbGaP study registration process. It's an NIH policy decision that right now that's part of the process. We are also about ways to help streamline that. I know it's a problem. But actually, the process for us in terms of making the data available is, especially in the newer studies, and some of the COVID studies that were mentioned, the PI conducting the study still needs to register the study into that, but the data actually doesn't get uploaded. We put it straight into the NIH control file buckets, which then they give it their own buckets. So we do so much good for that study registration process. Because we have such close collaboration with dbGaP and the NIH DACs, if you find yourself stuck, we are actually able to kind of mention and make your cases and expedite things. But you know, right now that study registration process is still kind of the beginning step that's required to get here. I would say one we'll hear more later this afternoon, but within the secure workspaces, if you have data that you produce or that you have access to by some other means, like have a BioData fellow who has access to BioData data. You can upload it for your own use within the secure workspace, but it's not going to show up in any of the search and browse that can be seen by any of the users.
Taking a step back to the 3 ways to check your access to data, can you help us contrast why you would use one versus another? Getting a little bit of a deep dive into the picture, are there comparative advantages to using one approach?
Yeah, absolutely. So I would say the first method, so the BioData Catalyst website method, that just gives you a list of the study projects and consents that you have access to. So no additional metadata, just if you're curious what consents you have access to, that's a good place to start. If you're interested in more of the file level exploration and maybe some quick summary statistics like demographics, not summary statistics, more counts on demographics like gender, racial breakdown. Gen 3 is a great resource for that. And if you're interested in the files themselves, because in Gen 3, you can actually get the files and look at those. In Picture, it's a good resource if you're interested in more of the variable level. So this just provides a summary. I think you can also find this information in Gen 3, but what I'm about to show you with the open access exploration is more of the value proposition for Picture, where you can type the variable and kind of retrieve aggregate counts based on filtering to a cohort. Does that kind of help a little bit differentiate? Okay. Awesome. These are great questions. Oh, I think we've got one over here as well. I think in the blue.
So in the open access tab, it says these don't require any authorization, but if you click explore now, and if you haven't already gotten access to, you actually just come up with a link to file a BioData application. Am I missing something, or is there supposed to be some data?
Yeah, so the data that's available here is aggregate counts. So open access is more helpful for, I can actually go to the next slide. This is the next part.
I can see that.
This?
There's nothing, yeah, because all I can do is if you put my request and it takes you to maybe BioData.
I see.
Lisa's going to do some laptop troubleshooting for us. Does anybody have some questions?
All right, awesome. Great, so kind of moving on to the next part of Picture then is this Picture open access. So as I was saying, the open access provides an intuitive kind of Google-like experience to search variables of interest and retrieve aggregate counts for each study. So remember, because you don't have authorization to access these data sets, you aren't able to access the patient level information of these studies. However, you can retrieve counts, aggregate counts based on your cohort to kind of get an idea of what studies could be useful for your research. So let me go ahead and pull up the demo here. All right, so to get to open access from the landing page, there are 2 ways to do this. You can either click the Open Access tab at the top or click the Explore Now button under Open Access. And this is the page that you should see when you do that. And so here you can see that this is the total number of participants that I have access to query on. And you can see that there is a ±3, and this is because we need to obfuscate the data to protect patient level information, since people do not have access to this data. So let's say I'm a researcher and I'm interested in height traits. in different populations. So I can search height as my variable of interest. So here I can see a list of the different studies that have some variable that matches my search term, which was height. This first tab, the harmonized variables, if you remember, I discussed the TopMed harmonized data set. So that is what the harmonized variables refer to. It was this TopMed effort to harmonize several data sets. And so I see that TopMed, the harmonized data set, has a body height at baseline variable, which sounds really promising for my research. So I'm going to click on that. And then I have the opportunity to kind of filter or select what heights I am interested in. But because I'm just interested in any height, I'm going to go ahead and not filter by any value. So I can click this and say any value. So give me any value for height. When I click run, that part of my query is added to my overall query, and I can see that the total number of participants has been changed. And this means that there are about 228,000 participants that have some data for this body height at baseline variable in the harmonized data set.
You're doing great. I'm just gonna zoom in a bit.
Yeah, sure.
If I go down. Should work. That's all right.
Can people see that okay?
Let's see. If it doesn't work,
Okay.
then I'll.
Yeah, that's fine. Additionally, I can build onto this query. So let's say I'm only interested in height variables for adults. So maybe I want to filter by age. So I can search age at measurement of height, because I happen to know this variable. So I can see that this comes up for the DCC harmonized data set, this age at measurement of height baseline 1. And because I'm only interested in adults, let's filter to say 18 years or older. I can see here that my minimum is 2 and my maximum is 100, which is helpful information for filtering. So I'm going to change this to greater than or equal to and type 18 and click run. Again, my total number of participants has changed. So this is the number of participants that are 18 years or older and have a height value. And so just for fun, let's say I want only female participants. I can click female, and again, my number is updated. So over here, again, Elisa has access to all of these studies, but if you did not have access to these studies, you would see a request button next to these study acronyms. And that would again take you to that dbGaP page where you can submit a formal data access request. You can also see different consent groups in these drop-down buttons. If there are between 1 and 9 participants in a cohort for your query, we obfuscate that information by saying less than 10. So yeah, this helps you figure out, which studies, which consent groups in those studies might be helpful for your research. Are there any questions so far? That pretty much sums up open access. Yeah, we've got a question.
So, is the question, is there like a data dictionary type feature here, or do you kind of have some idea of the variable name that you're interested in?
Yeah, so we use the data dictionaries to figure out what the human readable name of the variable is. So I know sometimes variables, right, could be gender, or maybe that's not a good example, like A1 could be a variable name, but that's not very informative. So we use that data dictionary to decode that information. So that way then it's, gender or body height at measurement 1 or something like that. So no, there is no data dictionary, but we use the human readable format. So using the name that you're interested in or a keyword of a variable you're interested in should work for finding those variables, if that makes sense. Awesome. All right, great. So the last part of picture then is authorized access. And so just as a reminder, this is when a user has access to at least one study from dbGaP, and this allows the user to query the studies that they are authorized to access and actually export selected data into an analysis workspace to really start that research. So I'll go ahead and pull up authorized access quickly. So again, there are two ways to navigate to authorized access, either the Explore Now button or the Authorized Access tab at the top. And you can see the format is very similar to open access, but because we have access to these data sets, the data is not obfuscated by the ±3, because we are able to access that patient level, participant level data. So I'm going to go ahead and build the same query that I did in open access, just with authorized access, but you can search any variable of interest if you have access to data. So again, I'll say any value for height, and then say age. Greater than or equal to 18. And also females. So I've built my query for my cohort, and I can see the total number of participants that are in this cohort. And when I'm ready to go, I can click Select Data for Export. Which returns this data tree. And so because I'm interested in the DCC harmonized data set, I can actually see the categories of data that I'm interested in and really get down to the different variables and the variable level of the data. So if I'm interested in, let's say, all baseline common covariates, I can check that. And maybe demographics as well, since I've filtered on age and female. And then I'm able to click export data to begin that data export process, which I'll go over in a little bit. But that's sort of the functionality of authorized access. It's very similar to open access, just a few changes. Yes, a question? Yes. Yes. So right now, the way that picture is set up, the queries are study specific. So you might have to build different queries in different studies, but you will be able to export it into a single analysis workspace in a similar format, which will be very helpful in starting that merge process. Yep. All right. Oh. All right, so now that we've kind of touched on all parts of the UI, the data access dashboard, open access, and authorized access, we can start talking about the API, the application programming interface. And so the Picture API allows researchers to use Python or R to search and query at the variable and variant level and export data into a workspace. And so Picture team actually maintains examples of this that are designed to get users that maybe aren't so confident in their coding abilities to kind of get started in this API in either Python or R. So we have a public GitHub repository that I've linked on the slides here that you can check out. Additionally, we also have projects, or these picture API examples are on the 2 analysis workspaces on BioData Catalyst. So that is BioData Catalyst powered by Terra and BioData Catalyst powered by 7 Bridges. So finally, to export your data into a workspace, we utilize what is called a dataset ID. And you can use this dataset ID to export your selected data into a workspace. And so this data is saved as a data frame, which can then be used in further analysis, either in your Jupyter notebook or whatever format you prefer. And so hopefully this demo will work with the computer, but I would love to show you how that would be accomplished with 7 Bridges here. So let me grab a link quickly. All right, so on the BioData Catalyst page, you can see resources and services. And here is where the analysis workspace pieces of the ecosystem are. So here I will go ahead and launch BioData Catalyst powered by 7 Bridges, which we will actually be hearing from next. So again, I will log in with my ERA Commons. So here we actually have the Genesis tutorial pulled up, which we will be going through later this afternoon. But if we come up here into the public projects, we can see that the Picture API has their own public projects. So this is where the coding examples or the API examples are to get you started using the API for Picture. So in these examples, let's see if I can get a preview for this 101 notebook. Yes, so as you can see, this is a really user-friendly notebook that walks you through how to connect to Picture, what you need, what it is, different resources to learn more, setting up your environment, and step-by-step examples with code. But if I scroll to the bottom here, so this shows how to use Picture functionality. But scrolling to the bottom here, we can see retrieving the data. So actually here, I will copy and paste my query ID or dataset ID that I got from exporting the data before and put it into this notebook, and that will bring in the data frame. So there is this, I'm not going to walk through it right now because I think that will be too much time and maybe out of scope, but if you're interested, there is a resource that teaches you how to use this dataset ID. And I'd be happy to discuss more afterwards with you. Okay, we've got a question over here.
Just want to backtrack and clarify something. So
Yep.
we query our data, we know what data is available, we select it, and we have full access. What is actually exported? Is that the individual level data exported to your workspace, and then you can carry out transactions?
Exactly, yeah. So what will be exported is a data frame where each row is a patient or a participant, and each column is a variable that you've selected for export. So when you went through that data tree and check marked the different variables, that will be each column.
So does this really mean harmonized interface system? Because let's say you're working with proteomics data, that it's having a harmonized data across we wouldn't necessarily be able to do this specifically. Let's say I want to run the GLOS for a protein, but I'd be able to access.
So in order to the data that is access to Picture and the other components of the BioData Catalyst ecosystem are those data sets in the BioData extension that has been ingested into BioData. So if the protein data has gone this ingestion process, and is one of the data sets available in the BioData Catalyst, the answer is yes. But if it's not, it hasn't gone through this ingestion process. We can reveal
So I have a specific kind of data use question, that's specific type of data, and that's mass spec based data. It's happening for potentially probably. So there's two types of information that can go in. One is you have an analyte X and a semi-quantitative value mass spec. It's very easy. The other data is the primary aspect, which is incredibly important for validating specificity of the field, also to figure out this dark matter. Things that we characterize what we're familiar with. So it's information we like to put up. The issue is that there's no place to do that. BioData can't do that. The issue of our NIH sites that allow you to do this, such as the Capital Workbench, it's fantastic. The problem is that you can't, there's no fire. You want to, of course, they want to see all the primary data and all the individuals. They can't do that. So anyway, the point is, will there eventually be a possibility, I would strongly suggest, of having a space for one to primary aspect behind the fire?
We try to answer that question if I can. So one of our focuses right now is sort of the system for a lot of downstream is absolutely bringing in multi-unit data and all the functionality that requires. So what right now today, if you wanted to say do an analysis in the NHBLI bench and then take your results from some actual product, put it into your private workspace and start pulling in complementary costs and do an analysis to know that. We have some proteomic workflows available as analysis takes there's no currently, if we don't currently have any mass spec hosted data. Yeah, no, that's good, but definitely getting more multi-omic. I think we would really want to talk to you to capture the use case really well so that we can understand sort of sticking in our pipeline. And that's one of the ways we work with future slider.
I think we've got some questions. Yeah, over here.
Just to reiterate what Rob said, this is really important concern for a lot of related omics data to top that's not generated by top data. And I think one of the most important things that Rob was mentioning was the controlled access, which is now becoming, a big deal for a lot of our parent institutions. So having some sort of assurances that
I'll comment that we are going to talk about bringing your own data in the next little section. So if you're allowed to upload data to
So it's good to have the option to select variable by variable study, but I hope there is also an option to select all, because I can imagine both like and also in terms of importing data, the data set ID, I hope I can select 40 steps, right?
Yes, so as you can see, because it's in that kind of nested hierarchy structure, you can select top levels. So in this way, by clicking this checkbox, I selected all variables in the ARIC cohort. So I think that's exactly what you were saying, right?
Yes.
Yeah. Yes, so because the variables are data specific, so you'll have to build a cohort for the ARIC, or sorry, you'll have to build a query specific to the ARIC study using ARIC variables. So you will need a data set ID for each study that you're authorized to access.
Right, but when you were working in the DPR,
Sorry?
you were copying the API command that you're copying,
Mhm.
can I put several IDs in?
Yes.
Okay,
Yep.
thank you so much.
Yeah, absolutely.
I'll make two comments in case it's helpful. So it is in our roadmap going forward next year with the less expensive compensation, certain free compensation, if you will, that kind of highlights key variables across the suite of data that are sort of related, which is, I think, a little bit of a and then just to the previous on that, around managing kind of governance for data sets that are not listed in the gap. We are working on a solution with NIH, this is our policy about this, to be able to use access controls that aren't just in the gap. So if the consortium has some kind of list, that we can see their data and maybe that's how it's going to be. It is in our roadmap to be able to still provide access to that data. Defer to that source for access to total life.
All right. Any other picture questions before we? Oh, yep. One more.
So when you export data, is that or separate at the same time? And another question is there a place where you can see all of the data exported?
A place where you can see all of the data that you've exported. I think the API would be a good solution for you. So if you build queries in the API, you can check out those data frames that you export. And that way you can see the data more. Is that what you mean? Or maybe I'm misunderstanding your question.
I think it's related to capturing all of the data sets you needed. It's a list of all the different data sets that you created over time. Is there also the API?
Not currently. I think the way to do that would be to save that in your workspace. So in the Jupyter notebook, you know, as you're working on it and copying and pasting over those data set IDs, then you would want to save that workspace and that would be your running list. But yep, yes.
And then the other question, the way you access data from the 700 business management is very similar. So we can access it using this as a data set ID. You can use it on all platforms.
Yep. Awesome. All right, well, thank you so much. I'll be available floating around. So if you have any further questions, don't hesitate to reach out. Thanks.
All right, fantastic. I love this discussion. This is great. There's a lot of energy in here. I'm Tony Pataludis, Program Manager at 7 Bridges. I'm going to fly through these next sections because a lot of this is also going to be covered in the demo this afternoon, but more interactively. These are kind of slides with the interface on it, and I can talk about things, but I think it's going to be more interesting later when you're hands-on playing around. And the one important piece that I do want to spend some time on is cloud costs, because I'm sure everyone in here has questions about what does it cost. But the bring your own data has been referenced a few times, specifically with proteomics there. So in order to talk about that, all right. Yes, so we have the flexibility to be able to bring your own data into the ecosystem. So we have the hosted data in an NHBLI cloud bucket that you can link to your project, but you can also upload your own data via a whole bunch of different ways. And again, this falls under data use agreements, limitations, and IRB policies and guidelines. We're all researchers here. I think we understand that. So in order to bring your own data in, you need to be in these seven bridges or BioData Catalyst powered by Terra environments. We're going to focus on the BioData Catalyst powered by 7 bridges environment right now. So that's what we're doing the demo in later. So when you log in, you can create these private secured workspaces. When you bring your data in, you and the people that you add with permissions are the only people that can access that data. 7 bridges cannot access your private data. So I want to put that right up front so everyone is aware. So you do not have to have concerns about whether or not we or other people in the BioData Catalyst Consortium are accessing your clinical data. It is your private data. You can work alone in the project. So again, if you put your data in that project, you're the only one with access, but you can also add collaborators. And we have a strict permission structure, which I'll go over in the demo, which allows you to set collaborators' permissions to whether they can read, write, execute, or have admin privileges to add others. You can also organize the data. So as you're uploading your proteomics and other omics data sets, you can organize them into file structures, just like your normal GUI environment. And so how do we get the data there? We have, as I said, lots of different ways. There's a command line interface that we have helped people install on their HPCs so they can send it straight back and forth from their institutional clusters. There's a file system. We have an API set up. Again, you can send it from your HPC. There's a drag and drop. We recommend not doing this for anything larger than 100 megabytes. It's just drag and drop into the browser. And if you have an FTP server, we can send it over FTP. Also, if you have a private cloud bucket, so maybe you have an AWS or Google Cloud bucket set up for your research group, you can link that. And again, you will be the only one, or your project, whoever has permissions, will be the only one to access that data in the private cloud bucket. So you can directly link that. And I think it was noted earlier, we've helped some researchers do that to access where they have controlled permissions, but it's not hosted already. We've worked with them and whoever had control over those cloud buckets to link the bucket so they can get access directly in the cloud environment. I mentioned you can create nested file structures, so you can organize your data however you'd like to have it organized. And there's the bring your own data. I'm going to demo this a little later, so we move through that very quickly. Next, we're going to move into tools. I'm going to go through that really quickly, spend a little more time on costs because that's what's important, and then turn it back to Elisa before we get to our research talk. So tools and workflows, that's where most of the demo is going to be spent. I talked about organizing your data in that same project file structure. You can organize your tools and your workflows and tasks, which are things that have completed, which are where you can find the output files. We have interactive analysis. So the analysis in the cloud is really broken up into two buckets: interactive analysis and tools and workflows. The interactive environments look exactly like they would on your local computers. We have RStudio, we have Jupyter Labs, which contains Python and Julia, and there's R in there as well, and we have SAS Studio. All of these environments also have a bash terminal where you have sudo privileges, so you can log in and install whatever you need to do to get your analysis going. It allows fast interactive prototyping. There's a bunch of different cloud instances depending on what kind of power you need for your prototyping. And we also have machine learning environments present there, such as TensorFlow. For the tools and workflow side, so the other side, you can think about these as submitting a job to a cluster almost. We have over 600 pre-parameterized CWL tools and workflows in the 7 Bridges environment. And we have a GUI editor. So if you are not comfortable writing code, you can take any of these 600, and there's a GUI drag and drop where you can link inputs, outputs, and build out your pipelines. As I said, over 600 fully parameterized, customizable. The vast majority of these also have benchmarking information, which is going to be very important as we move later on to the costs. It's a searchable interface. I just wanted to highlight some of the ones that are probably important or interesting to the audience here. So association and kinship testing. You can search up there. You can see I searched Genesis in the screenshot, and we have a whole bunch. We have a full Genesis pipeline there already pre-parameterized, as well as notebooks to help run that. You can also bring custom tools. So you search and you say, Tony, I looked at your 650 tools and I did not find what I want. So you can design it yourself. If you're comfortable coding, you can wrap your own tools with CWL, or you containerize it and then wrap it and upload it to the 7 Bridges environment. This is also where our interoperability that Elisa will cover with DocStore comes in, that you can go to DocStore, which is a tool registry, and search for CWL workflows and then pull them into 7 Bridges. So in addition to having ours, you can look for workflows in DocStore, or you can design your own if you're comfortable, and we're happy to work with you to design your own. What the workflows allow you to do is really scale up your analysis. So maybe you want to look at 50,000 whole genomes. That's going to be pretty computationally intensive to do multivariate association. So you can use something called batching and scattering, where you can put all of them in and scatter it across the cloud. And all of these can run in parallel, and I'll show you how to be able to track those tasks. They can all run in parallel, which means your computation goes faster, you get your results faster. We have detailed documentation and tutorial around all of this. There's a 7 Bridges Knowledge Center, which is linked here. If you're looking at the slide deck, you can click that bright red button and look through all of our documentation and tutorials. We also have 24/7 support on all the pages of the 7 Bridges platform. There is a wonderful question mark down in that corner. You click it, it'll pop up and say, what do you need? Questions on bring your own data and tools? I know I went really fast. We can cover this. I'll be around all week.
So on tool development, how much do you do and how much do we have to do? For example, you gave an example of a parallel computation, which you would call obviously parallel. 50,000 sequences put on 50,000 computers. What happens when you have a computation parts, for example, a terabyte of RAM?
So I think there's 2 things here. So the tool development, there's a question around, and then the batching and scattering. And I think maybe they're linked right now, but let me talk about them separately. So in the tool development, say there's something new that you want to run, just use FastQC. We have that on there, but if you want to check your reads and it wasn't there, you can submit that to 7 Bridges and we'll put it in our backlog as a user requested tool that we can build out and put out that CWL tool. I think that was maybe the first half of the question. The second half, your question was around the resources and the batching and scattering.
What's in the back? And how do I access it? So in other words, how do I get a larger computer?
Yep. So there's so in the cloud, there's Google and Amazon have things called instances, and they're kind of like nodes. So if you've worked on like a cluster before, or if you haven't, it's like an individual computer with resources, and you can tell it how much of that you need, and it will aggregate more of those together. So in this wonderful place called the cloud, whatever nebulous place it is, you can request and scale up the amount of resources that you need.
So I developed the tool to request that.
So for the pre-built tools that we have, you can go in and say, I need additional resources. Maybe I can cover this in the demo, but you can select what kind of resources you need, and it will interpret.
You have large computers with large RAM.
Yes. Yes.
You don't have to be able to code to access that. You can say, I need something with these characteristics in the GUI, but you make it happen. If you run up into
Sorry.
that's where, I think Tony can show you in the demo, but potentially accessing that help and say, I need something more different than one of your kind of presented options. And that is an example where we, in the machine learning pipeline that was mentioned earlier, we actually worked with that community to get the kind of compute we did before that.
It's a good example. It's not just a request 10,000 computers for RAM.
Yeah, exactly. Working with Tony, working with and others, with the CFPG folks who are running kind of neural networks against large biological approaches. That was an example where we actually needed to add some additional options so that they could use that.
Another question on the, since this is cloud, there's a little checkbox in the app. Are you upload this to the cloud? Do I assume that you've done that, or do I have to check that?
One of the hosted data sets, so something that you can see in the system, you're not creating it yourself. We've taken care of that for you in the together. So question and scattering is one different from saving time, but does it save me money or cost me more?
A great question. And that all depends on your specific case. So I know some universities, some places charge for storage. The HPCs that I've worked on, I was at the NIH, I was at the University of Connecticut, they didn't charge for compute. And it also depends on what indirect costs for your institution look like, right? So like a lot of universities, HPCs will be supported through indirect costs where the university is, maybe there's overhead on a grant and they're supporting the infrastructure there. And so maybe it's a hidden cost, but that cost still exists because you still need storage, you still need computers. I guess the question about whether it saves you money or not is a complicated question. We have, as I have a presentation here, we have ways to optimize what you're doing in the cloud to try and save you costs as well, if you're working in the cloud. I think that's probably my best answer. Becky, I don't know if you have anything to add to that.
The issue of cloud costs is complicated, so we need to do our best to walk through it. So, what resources do you have for free and non-charges largely depend on the situation, as I said. Within the system, we also have credits, which is NIH and providing funds, so free that cover credits to cover your compute costs up to a certain level. It's very easy to access those. It does take us a few days to process. We have to get NIH cost. We have gotten that request down to a couple of days to get a response to get those provisions. And Tony will show you there are options within the system where you can basically select and configure machines that are going to run it maybe more cheaply, but maybe you'd rather spend money to work them faster. And I think that's part of it.
That's exactly where I'm heading. All right, we're going to run through the cost real quick. We'll take a really short break and then Ravi will talk, right? Cool. All right, so to your point about costs, understanding, estimating, managing cloud costs. We have a wonderful agenda here of what are the costs, how do you estimate them, and what potential funding is available, or how can the BioData Catalyst Consortium help you with that funding. So cloud costs can be broken down into really 3 buckets here: storage, compute, and egress. We're going to focus on storage compute because those are the most important and potentially the most confusing. There is a web resource here, a link again, about what cloud costs are and what cloud credits are. It's important to note, again, the hosted data that Emily had gone over, that we've talked about a lot, you are not charged for that. So even when you bring it into your workspace, the original hosted data, not the files downstream of them, but the original hosted data, you will not be charged for storing. They are paid for by the NIH or supported by the NIH. So to estimate your costs, as I said, there's kind of a couple different buckets here. There's data storage, computing and analysis, and egress. We have a guide on the Seven Bridges Knowledge Center to walk you through this process as well. Data storage, all of this is direct pass-through. So data storage, you can go to Amazon or go to Google Cloud and look up how much does it cost. I know standard storage in Amazon right now is 2.3 cents per gigabyte per month. And so for that's only for data that you bring, or again, derived files, output files. So this includes any data that you have uploaded to the platform. As I have noted numerous times, it does not include the controlled data sets. So those CRAM files or large files that NHBLI is hosting, large genome files, you do not pay for. The cost will vary by the amount of data you store. As I said, it is on a per gigabyte per month basis. We also have archiving enabled on the Seven Bridges platform. So if you're not going to touch files for a while, you can put them in archiving and the cost is drastically reduced. For compute and analysis, there's a range of factors that affect the cost, which is why, as Becky said and I said, it gets kind of complicated, but we're going to do our best to try to help you calculate. Has to do with the platform and the cloud infrastructure. So as I was mentioning, when you go to compute, there's a whole bunch of like mini computers in the cloud, right? And based on how many of those you need and what kind you need, what kind of resources you need, the price per hour varies a bit. But ultimately, you're looking at CPU hours. So it depends on the workspace and the length that takes to complete. And then egress charges. Egress is when you move data from the cloud or to a different cloud, right? So if you're downloading, say you upload your data to a cloud bucket, when you download that, you get charged egress. Or if you're moving data from Google to Amazon, there is an egress fee. You can plan costs ahead of time though. So if you want to put this into your grant, again, we're researchers here, based on grants, you want to put this in, you can plan for it. We have lots of benchmarking information for a lot of the apps that I had talked about, the CWL workflows that are pre-built. Okay, this is, I know there's a lot of stuff on the screen. There is a guide here with detailed benchmark CSVs based on the number of samples input for each of the tools in the Genesis Association testing pipeline. To manage your costs, you can take that benchmarking information and then in the platform, as you run tasks, you can view and compare it back to what the tasks actually cost. So here, for instance, is the single variant association test, GDS conversion, and null model fitting. You can see the instance, so that's the compute in the cloud that you needed, cost 21 cents per hour. Attached disks, so storage, cost 3 cents. So the entire cost for whatever this run that someone had run was 24 cents. We have a billing page on the platform. So when you create an account, you get a billing group and you can check your billing costs broken down by compute for both the interactive sessions and for the tasks run. And you can check your storage costs. And I'll walk through this again in the demo. To fund this, as Becky noted, we have NHLBI supports for HLBS heart, lung, blood, sleep researchers, pilot credits. So if you go onto their website, the BioData Catalyst website, you can request up to $500 in pilot credits to pilot your experiments, try out the ecosystem, and figure out how you like it. That can be either divided or directed towards BioData Catalyst powered by 7 Bridges or BioData Catalyst powered by Terra. Credits workflow, you should sign up for the community, sign up for one of the workspaces such as BioData Catalyst powered by 7 Bridges, apply for the pilot credits, and after you run the analyses and you figure out what this might cost you, you can apply to NHLBI for additional credits. There's no guarantee, but you can apply, you can request additional credits based on the research that you're doing. We also have put a guide together and recommended language to put into a grant. So to begin with that, you would begin if you are itemizing your grant, you'd begin by calculating anticipated storage cost, what your compute might be. We have recommended sample text, and there's a link here again in the big red button to this article. And we will offer letters of support from the BioData Catalyst Coordinating Center. So at that, we can take some questions or take a break.
Sorry for fast, so I didn't catch you talking about the post. When I submit a shot, do I see the post before I run it or after?
The cost will come after you run it. So because of how jobs are submitted, there's benchmarking information, so you can try to predict about what that job will cost. But we don't have a way right now to predict before you run it, because it's based on the type of data that goes in, what kind of analysis is being run, what kind of compute resources, right? It's a large multifactorial problem. This is something that we're working on though.
I'm really impatient, but also cheap. Are the number of instances directly proportional to the cost? Or is it more complicated?
I would say resources are related to cost, right? So if you need more CPUs, it is likely to cost you more, but I wouldn't say it's linear that, right, yeah. But we do display the costs per instances there, so you can look and find the 12 cents per hour instance for your compute. Another question behind you.
You mentioned that you must have a data Genesis is one. Do you have a contact for other software, for example, the in-house?
So Genesis is one, and you want to know if there are other association tools present. Yep. Yep. Plank and EPAX are on here. Star, I believe we're working on if it hasn't been released yet. Genesis is on there. Plank is on there. We have GATK tools released on there as well, I believe.
Do you have a cost estimate? For example, we are using the EPAS, do you have a table, like what you showed?
Like for Genesis. So I would have to check. I can incorporate that into the demo. So in this public apps gallery, you can search for it. And when you click into the tool, which I'll show you, it'll give you benchmarking information that we have.
Could you say a bit more about the relationship between billing group and workstation projects and user? And what is the hierarchy? And a related question, the PI is such a what control do we have over those layers? Let's say one user does it over.
Yep. So you can create a user pays billing group as a PI. You can email and get that and have admin privileges, and you would control who could get added to that, or you could add on another admin. Maybe you have a lab manager that you want to add as an admin to add people. And you can control who is on it and who is not. When you create a new project, it will ask you for a billing group. So the project, you can select which billing group you want. And we've had PIs that will make multiple billing groups for their different grants, right? So they can box out their spending appropriately. When you view the spend, the spend is broken down by project. So you could have 5 projects all to one billing group, but then you can still see what spend is broken down based on those projects. Following through on your questions. When we create a billing group, we'll ask you for a maximum cap, and our support team will track what that cost is. So we typically recommend $500 or $1,000 to start. And you can watch that in real time, how it changes. But we'll send you an email as it approaches, probably around, if it's $1,000, around $800, and say, hey, this is where you're at. Would you like to increase it? We have guardrails in place so that when it goes over that cap, if there's an analysis running, it will continue running, but you can't start anything new. And we will, again, our support's really on top of it, and they'll reach out to you immediately. Were there other questions there? I might not have hit all of them.
We got a thumbs up. All right, other questions before break? I think at this point, Tony, we're just gonna take out like a stretch break 5 minutes. Feel free, if you have a very specific question, the background will start to break. There are comments in the back of the room, and they're just like light finger food and snacks that are still outside. So we will reconvene, just after quarter two.
Folks on Zoom, we're taking a 5-minute quick break to stretch.
Yes,
And I can point out parts of
that's the
that
coolest
they've
part.
heard.
Yep.
Um,
All right, we're gonna get started in just a minute. I'll just go like this. And then down to, it's right even right there. I guess I go side down there. Do you want speaker notes or that anything?
What?
Do you want the speaker notes at all?
I think I have, actually.
Okay, so then, yeah, it's pretty straightforward.
Yeah, that's fine.
And forth. Yeah, just
Yeah. just- I'll look up
What
how
are
to,
these?
No, it doesn't work. It's not plugged in. There's no USB.
Well, no, I was just using the. Can you see
Oh, the
that?
pointer.
Tony, can you see this? This you can? What? Can you make it bigger? I don't know.
Just make sure you're a little closer to the microphone than Becky was. There's
Okay.
a little bit of feedback.
Gotcha.
So Tony stood a little closer to the mic,
Okay.
which allowed like just the sound altogether was better. All right.
No. Okay.
So
I
just use
won't
the
it.
mouse.
Yeah.
You can- if you move the mouse over to this side, oh no, this side, it shows up.
Yeah. I can just use the mouse.
You're showing up. How annoying. Well,
So the one that was sitting behind me was like, yeah, I can't really see that. So I would actually use the mouse
Yeah.
to point things out
trying to get,
because I think people will be the like pointer.
oh, there it is. I found it. That's so annoying. Oh, there it is. Ah, so it is this way. If you take the mouse and it goes over to this side, it'll show up.
Oh, it goes over there.
Okay.
It's a little weird at first, but once you get the hang of it, you're like, okay. Okay, awesome.
Cool. Thanks.
All right, we're going to get started now. Please get back in your seats.
If I can ask you guys to gather during some food and have a seat, so we can, while we can kind of walk you through some of the work piece we're doing on the BioData. I will say, you noticing that a lot of folks in the BioData team standing up and walking around, so if you guys feel like you need to stand up and stretch, please go ahead and do that. You don't need to stay seated. We're a casual lunch, but if you guys feel sort of rather nice.
All right. Thank you, everyone. So as Becky mentioned in the beginning, my name is Ravi Mather. I'm a research statistician at RTI International. And I was a cohort 2 BioData Catalyst Fellow. And what my title of using TopMed whole genome sequencing data as public controls for case-only data, which I hope this gives you a use case of how I've kind of used a lot of the services and platforms that you've heard about and kind of synthesizing it together into a research project. And I'll go through some background, some results, and the method GAW-merge that I've developed with colleagues, along with some conclusions from my work in BioData Catalyst. So in the background, matching population or public controls to case-only cohorts is a standard epidemiological approach. And with this approach, you can control confounders and cut costs in your particular study. And particularly in dbGaP, there's 217 current studies that are case-only cohorts where there are no controls collected with that. And previous successful applications by my colleagues have these applications in GWAS data is particularly dependent on having a consistency in ancestry and a substantial overlap in genotyping variants across your cases and controls. But with the NHLBI TopMed cohort that Emily described earlier and its whole genome sequencing data, you can address both of these issues. So a little bit background of the TopMed Precision of Medicine Initiative. It's sponsored by the NHLBI. It provides integration of whole genome sequencing and other omic data, as has been mentioned in our discussion today. It's specifically heart, lung, blood, and sleep disorders. And all of the data, as Emily mentioned, is hosted on BioData Catalyst. So to go into the diversity of this TopMed cohort, and those familiar may be familiar with this, but as Emily mentioned, over 100,000 participants. And a big drive of this cohort and this initiative is 60% of the cohort is non-European. Thus, we're getting that diversity in ancestry. And what's shown on the left here is the phenotypes of interest with our heart, lung, blood, and sleep disorders established throughout the cohort. So with my population controls methods, questions I want to answer is, how do you select whole genome sequencing cohorts for case-only array data? How do you do that selection? And a lot of that is driven by picture and Gen3, as Emily mentioned. And the next three are questions that I'm looking at of how do you integrate array and whole genome sequencing data, and are these compatible? And then with that integration, can you control false positives and replicate true signals? And these last three are implemented with the protocol on the 7 bridges system. So with the analyzing the particular array data that we used, so to the left is the COPD gene cohort, which many may be familiar with. Sorry. So it's one of the largest studies to study COPD. It has just under 10,000 samples, and both African and European ancestry. And the array data is available in dbGaP, which in my application, I've used the bring your own data to upload this array data. So I've gained access through dbGaP and uploaded the array data using the 7 bridges system. And then to the right is the CoGen, which is a study of nicotine dependence, initiated in 2001 with collaborators Laura Beirut and Dana Hancock. It has nicotine-dependent cases and non-dependent smoking controls. It's from Detroit and St. Louis with over 2,900 blood samples with a human Omni 2.5 array, SNP array. It has both African and European ancestry individuals. So with the COPD gene and CoGen array data, how can we match as particular test cases of which whole genome sequencing data to match with this? What's really helpful in this case is, as Emily mentioned earlier, there's the TopMed harmonized data with the publication shown there on the right, where the DCC, the TopMed DCC has harmonized 63 variables for across 17 TopMed cohorts, and includes various different variables for atherosclerosis, sleep, inflammation, lipids, VTE, blood cell, and various others. And as Emily mentioned, it is hosted on BioData Catalyst and searchable in those platforms. So but a particular example here is looking in the Gen 3 platform that Emily mentioned. So both of the COPD gene and the cogen cohorts are smoking samples. So we wanted to get a COPD gene cohort with European and African American ancestry. So what you're seeing here is the Gen 3 platform. Sorry. Where I've filtered down to just the COPD gene individuals. It's looking at both European and African American, where you get the distribution to the right. And what's not shown, but it's further down on this page, is I've selected just those that are current or former smokers in this cohort. And another way to do it, which Emily did a great job of describing, is the API, which is available on the 7 Bridges system, as Emily has described already. And the same race and harmonized variables can be accessed that way. So with the Gen 3 and picture tools, as kind of has been described, if you can identify which cohorts do you want to integrate together as whole genome sequencing. So now to go to the next step is, how do you do that integration? And with looking at our array and whole genome sequencing data, one thing we wanted to test is, in doing this comparison, are we getting GWAS signals that are technical comparisons with the different technologies? So what we looked at is the COPD gene cohort is available with both the array data that is mentioned and is also part of TopMed. So we looked at the same samples that just have 2 different genotyping technologies with array and whole genome sequencing, having the technology as a control. And what you see on the right is a Manhattan plot that you rarely see, that there is no signal. Completely flat Manhattan plot to the right, where you're not getting any technical artifacts in that combination of whole genome sequencing and array data. So we have there proven that we can compare whole genome sequencing and array data with that comparison. And next, to look at with this integration, are we controlling for false positives in our genome-wide association study? And here we took all of the COPD cohort and matched it with the cogen cases. So here we have no case control count, since they are COPD gene and cogen are both smoking populations. We don't have a nicotine or a smoking signal showing up. And we do a meta-analysis using metal and using 15 principal components in both European and African-American ancestries. And since they're all smoking samples and we expect no signal, what you see in the Manhattan plot down here is there truly no genome-wide significant signals. Sorry. And the lambda with the QQ plot is controlled. So we are controlling for false positives. And so we have conducted that we are controlling the false positives with this integration protocol. And finally, we want to replicate known COPD signals. So this is the Manhattan plot from the Hobbs et al. Paper published in 2017, where they have many genome-wide significant signals throughout the genome. With the covariates indicated, and specifically the HHIP signal on chromosome 4 that's shown with the locus zoom plot on the bottom right. So we looked at, as we are integrating these data sets, can we replicate these signals previously identified? So here we're looking at the COPD array data, which is the nicotine data set with COPD cases. So since they're both smoking controls, that's controlled there. And then we have COPD cases only integrated into that data set. So the analysis is a meta-analysis again across both European and African ancestries with the first 15 principal components and as covariates across both African and European ancestries. What we see there is both on chromosome 4, the HHIP, which is the Hobbs et al. Strongest signal, that is replicated. We also get another signal with FAM13A, TFB2. Keep clicking, sorry. NCR, NA3, and CRIP2A6. So we are getting replicated signal. Therefore, this integration protocol is actually both we're controlling false positives, we don't have technical artifacts, and we can reproduce known biological signal. So how we talk about tools, so Tony talked about tools in development. How is all this analysis happening behind the scenes in the Seven Bridges platform? This is our workflow for what I'm calling GAW merge, which is genotyping, array, and whole genome sequencing merging. So it's a general protocol of integrating array and whole genome sequencing data, where we've shown here with the case-only samples. So specifically here at the top, this matching with the key features, this is where the Gen 3 in picture has been really integral in identifying what are those matches. And one big step that we've found is only having the overlap, as was mentioned in our previous work, is the overlap of the SNP data and the whole genome sequencing data is really important. So since we're doing that, kind of taking whole genome sequencing data and down to the array data, after quality control, we are phasing and imputing whole genome sequencing data. So we're taking it down to the array data and then imputing it up for whole genome. So with that, in the Seven Bridges platform, I've implemented both an imputation strategy as with Minimac4 and phasing with ShapeIt2 with workflows with both of those tools. This reimputation, what we found, there was some technical, there's a little bit of signal shown where that imputation up, there's slightly a different imputation, what's called an empirical R-squared, or how the imputation of your algorithm compares to already genotyped data. So we found those to be mismatching sometimes. So we removed any empirical R-squares less than 0.9. And after reimputation in this association test, Tony mentioned a couple others. I'll mention another one here that was implemented in this case, which is RV test, is rare variance test. So we have a workflow for that association test also. And finally, we have filtering of the final GWAS results. So as mentioned, this was all done in the CWL common workflow language in the Seven Bridges environment on BioData Catalyst. And so one thing that's nice with CWL that Tony kind of went over real quick, there is that GUI interface for it, which I don't know CWL at all. I cannot code it to save my life, but I can still create a CWL workflow with that GUI interface. So it's a really nice way to kind of create tools in a workflow. So in conclusion, I'm happy to take questions. With the identification, we can use Gen 3 and picture to identify what are the cohorts that we are integrating with the available array SNP data that is. And we can use, we've used Seven Bridges and its tools for developing novel tools and workflows. And as I mentioned, it's really easy way to conduct research. One thing I really like is it's always a headache to download data from dbGaP, decrypt it, store it, and do all that. All that headache is off your shoulders, which is really makes you focus right on the science. And as has been mentioned, being that like white hat type thing, kind of getting that resource is really nice. With that, I'd like to thank RTI International, my collaborators there, Eric Johnson, Dana, Nathan, many others, COPD, Gene, and Cogen for data availability, TopMed for creating this great data resource, and finally BioData Catalyst for the funding for the fellowship. Happy to take any questions.
Any question? Oh, we got Becky. We have a question over here.
Thanks for a great talk. So I guess, what is for a user using your workflow? What is that? What does it look like to the user? Or instructions?
Yeah, so I don't have a tool here, and I might go back to Tony's slide that shows the screenshot of it. Right. There it is. So I'm gonna borrow Tony's slide here of the workflow on the right, where you can kind of drag and drop these tools around. Each circle in this case is a different tool, and the arrows represent a input or output going to different tools. So with this kind of GUI setup and a network setup, you can say that you have your QC pipeline, and that is going into phasing, which is going into the imputation. And each of those have a different workflow behind it with the same kind of GUI interface. Does that make sense?
That makes sense. I guess I wonder, how do you ensure that the files that you're putting into this workflow are actually in the right format? Is there a backend where they'll tell you that the file is not appropriate?
Right. So for a lot of the BioData Catalyst or the Seven Bridges developed tools, there is that QC, and that's kind of developed in the original workflow. So if you have a Plink workflow, for example, inputting in a VCF file or a Plink formatted file, if you have something else, that will be identified, but it's on the tool developer itself to put those flags in as kind of QCing the input.
I read about that paper describing recently in that POS and in the slides. Do you have like a notebook that's ready to describe exactly?
Yeah, I do. So I do have a GitHub that I should have popped on the slides. I do have a GitHub that describes all of the methods in both. So all transparency, I have actually implemented this method in both BioData Catalyst and with other funding in a platform that's called DNA Nexus. I'm sorry if there's DNA Nexus folks in the room. I know it's a Seven Bridges competitor. BioData Catalyst is 10 times easier to use. So kind of comparing the two, is DNA Nexus is another cloud computing environment. But we've implemented this in both, and both of them are documented in our GitHub.
Don't worry about using the available. We're able to use it in both places and kind of quick and replicate results. Other questions?
Awesome. So I'm going to give a brief introduction to some of the interoperability features in the BioData Catalyst ecosystem there, and then I'm going to talk a little bit about the Genesis pipeline, and at that point, we're going to have an interactive session where you can be following along with Tony on BioData Catalyst powered by Seven Bridges in actually performing a genome-wide association analysis with some demo data. So I encourage you all to move forward at some point so that you can really be close enough to the screen to see what's happening. But let me pull up my slides first. My name is Elisa Manning. I'm from the Broad Institute and MGH, and I play a role in BioData Catalyst as one of the ecosystem component PIs in the consortium. So I'm really just happy to be with a crowd of people that I know, because I've been part of Charge for many, many years, and I love the Charge consortium. I really love our masks that have the logo on the side. It's better than a hat or a bag. So BioData Catalyst is an ecosystem of these platforms. And so the user flows through the different components will be determined or specialized for each user community. For example, the Charge community and the Charge Analysis Commons is being offloaded onto the Seven Bridges platform. So we're focusing the discussion today and most of the demos today on Seven Bridges. But at each stage, there are multiple components available to platforms. For example, exploring data on either Gen 3 or Picture, doing analysis on Seven Bridges and Terra, or using the doc store or the TopMed imputation server. So our goal is to have many services available to the NHLBI research community so that to enable their research in the cloud. So we've actually already gotten an introduction to the Gen 3 platform, but one focus that I'll bring your attention to is that the data files that are available to you in the Gen 3 platform have a hierarchy and data tags or metadata associated with them, which allows for very rapid filtering and searching of files. When we actually did see this, I think earlier when Emily was up here. So the interoperability feature that I'm going to show you is how to select files in Gen 3 and then export them to a workspace in the Terra platform. And it's a video that I'm going to show you just to give you a sense of how the Terra platform works. It's very much similar to the Seven Bridges platform, but it's not identical. There are a few differentiators. For example, our workflow language is the WDL workflow language, not the CWL workflow language. And there are pluses and minuses to each of these workflow languages. We like to say that WDL is readable. So it's something that if you feel comfortable on a Linux system on the command line, you could easily translate your work into WDL. We work on the Google Cloud platform, and soon we'll be on the Microsoft Azure platform. And we have some preloaded applications like Galaxy, the Genome Viewer, and then the Seeker app that you might be interested in. Our interactive analyses much like Seven Bridges, feature highly customized machines with persistent disk sets, that basically a persistent disk that you can keep data in as you reconfigure the cloud resource that you'd like to use. We have Bioconductor, the Hail Toolkit, GATK, and other bioinformatics tools. And like Seven Bridges, there are these best practices workspaces that are from tool developers or from user communities focused on getting researchers up and running, doing their research as quickly as possible. So the video I'm going to show you is, I'm going to get it started, and I'm going to fast forward through it. So this is an example of starting on the BioData Catalyst webpage, launching BioData Catalyst powered by Gen 3, going to the Exploration tab, and selecting File rather than Data, because we want to export some files in the same organization that they are here in Gen 3 into a Terra workspace. And we'll get to a point where we will see what that looks like in the Terra workspace. So I'm actually selecting TopMed FreeZate data from 2 different cohorts. I think I chose the Amish cohort and the Mesa cohort. So you can see I've gone to the appropriate piece of metadata, and I'm searching and filtering and selecting. Okay, so I'm not going to. So I'm at a point where I've selected 69 VCF files. And those are the files I want to download to my workspace. Oh, on the bottom, it says your export is in progress. So it does take a couple seconds for the export to complete. When it finishes, you're given an option of where you want to export it to on the Terra system. So I had gotten to this point by clicking the button up there that says export to Terra. So once you get into that, you can say create a new workspace in Terra, and then you can enter the workspace name, select the billing group for your new workspace. If you're using user groups and you want to restrict access to the data on Terra, you can create what's called an authorization domain. It works very much the same way that Tony explained for Seven Bridges. And then within just a few minutes, you're brought to a Terra workspace where you can look at the data organization. So you can see there's data being exported successfully. And then I show an example of going and learning more about how the data is organized from Gen 3 in an analysis workspace. Oh yeah, and now I'm just going to take a quick look at the tables, refreshing the screen. And now you see there are multiple tables, and the files that I've exported are organized in a particular structure, which is defined by how they're organized in Gen 3, linked together through this metadata structure. All right, so I'm now going to very briefly introduce DocStore. So this is an app store for bioinformatics. Users can launch workflows from DocStore directly into Seven Bridges or Terra, or they can download them to use locally. So DocStore has a command line interface that you can use to quickly run workflows on small, tiny data sets or toy data sets. So having a place like DocStore to put our workflows has been really beneficial to my research lab and our researchers that I work with, because workflows are versioned, they can be updated automatically from a GitHub repository, and they can be easily exported into one of the workspaces. And we can publish workflows on DocStore. So here's a good example of a workflow that is being cited with a DOI in an article. And that brings to a standing page that is supposed to stick around for a long time, so that it have a good record of the workflow as it was published. So this video will show us how the export process works. So here I'm in a Terra workspace, and I've gone to the workflow sections of the workspace and said, find a workflow. I selected doc store as the source. And I'm at doc store and I'm searching for the workflow I'm interested in looking at. I find it, and then I can look at the workflow and find out various pieces of information. And I can launch it, as you can see on the right-hand side, with one of these cloud platforms. And when you do so, it asks if you select Terra, you're asked what workspace do you want to put this workflow in, and you're given a list of the available workspaces for you that you either own or have write access to. So then we're just exploring DocStore a little bit. You can read the actual code underlying the workflow, which might be useful. Here I'm showing how the particular tool is readable. Now I've exported to Terra. Once again, in this instance, you see the whittle text as you're doing the import. You've chosen your destination workspace, and now we're in Terra where we can configure the inputs to that particular workflow using a number of different functions. Good. So I will do the intro and then pass it over to you. Okay, so just in case people are not familiar with the Genesis workflow or the tools that we use to implement a genome-wide association study, I'm going to give a brief introduction about the underlying code used by the pipelines and workflows that we'll be demoing and practicing with today. So the TopMed analysis pipeline is a package distributed by University of Washington, the GAC, which was the group that was the TopMed coordinating center. The code that they have has many analysis steps, and you can actually go to this GitHub repository and read documentation about the components of the pipeline. So the analysis steps include converting a VCF file to a GDS file, which is a different file format for genomic data that is a fast format and therefore is used by this particular set of packages. There are functionalities for determining relatedness in your data set and generating principal components for population structure and further adjustments in your genetic association tests. There's functions to create a genetic relationship matrix from the data in a GDS. And if you're not familiar with what that means, it's just another one of the tools that we use in our genetic association analyses. And the various types of association testing, there are 3 types that I'm going to describe: the null model step, a single variant, or a rare variant association tests. So this pipeline has the first step of this genetic association steps is what we call null model, the null model step. And we throw that term around a lot in our community without realizing that it's a completely made up name that only those of us who've used this tool or read the paper would know about. And so the null model step leverages the phenotype data that you plan to use in your analysis. So basically the phenotype file with the columns of the outcome and the covariates that you would be using in your model for your genetic analysis. It might also use a genetic relationship matrix if you're correcting for population or a family structure within your data set. But basically it takes everything, all the pieces of data that you have compiled that are not the genetic data itself, and runs an operation on it to get those data ready for a genome-wide scan. And the operation is basically looking for the association, basically fitting a model under the null hypothesis, which means no genetic association. So this step puts the data in a format so that it's, it does a lot of pre-calculations on the data so that when you get to the next step, which is either a single variant association test or a rare variant association tests, The software then just leverages the data that exists and does a scan across the genome. So you're basically simplifying the number of analysis steps that need to be performed for each test in your genome-wide association study. Like by pre-computing a lot of the operations, the piece that really depends on the genetic variant happens really quickly. And since we study millions of variants, we want our analysis to happen quickly. There is this tutorial project on the BioData Catalyst powered by 7 Bridges that we'll be going through today. This text is basically the intro text to the workflow. It's designed to introduce the user to the Genesis R package and related R packages. There's an interactive analysis, but there are also workflows. So this tutorial project really does cover a lot of the bases when it comes to doing analysis on one of our platforms. And Tony, I think I'll turn it over to you and you're gonna lead everyone through.
Sure.
Okay.
Thank you.
Sure.
All right, so I wanted to break the demo up into 3 sections. One is kind of the introduction to the 7 Bridges environment. How do we make a project? How do we find the resources that we need? How do we add people to our project if we want to expand, add in our lab? So we'll go to BioData Catalyst, services, and if I scroll down. We will launch 7 Bridges. I'm not logged in. I'll log in with my ERA Commons. If I remember my password. Okay, and just as before, so you can see we log in with the ERA Commons. I can authorize, and my account has no access to control data. This is my test account, not my developer account. I'm already in a notebook here. So when you log in, that's because I was already on that page. It saves where I was at. When you initially log in, which you can do right now if you want, you can follow along with me. This is the screen that you'll see. And so to orient you here, up across the top, there are projects, data, some galleries, and public projects. All the projects that you own or you are a part of will appear over here in a list. And if there were more than 10 or so, you would be able to flip through that list. Over on this side, you can see tasks that you've run or interactive analyses that you've run. So you can see
I was just going to zoom in a bit.
Oh. You can see different notebooks that you've run before, or you can see tasks are the CWL workflows and tools. And so the first thing you probably want to do, you get on the platform, you want to create a new project. Before I dive into that, if you have your ERA Commons ID and you have not gotten an email that you are added to a billing group, please go to the forum that we had posted before, which you can access from that BioData Catalyst main page. And there should be a topic there for getting added to that billing group. And so that's so you can run analyses with me during this workshop session. My colleague, our community engagement manager, Dave Robertson, is on Zoom and on the forum for anyone who has not been added to that yet. So if I want to add a new project, I can click into projects here. And down at the bottom, there is a button with a plus sign. Can't scroll down, but if we click that, new project comes up. Everyone that has an account by default gets Pilot Funds account. And when you apply for your Pilot Cloud credits of that $500 in credits, it'll be applied to that account. Unless if you applied before coming today, you will not have any funds in that account. So you need to use something else. Oh, that was not what I wanted to do. Sorry, now I'm getting lost. Let's relaunch. There we go. All right. So I wanted to create a new project. And when you go to create a new project, there will be a pop-up here. You can name it whatever you want, and that'll give it the unique URL. So I can name this test for charge. And you can see down here my user ID and test charge here. So this, if you have someone added to the project, you can send them this URL and they can access it. Only, again, if they have permissions to be added to that project. I'm going to zoom back out because I am failing at scrolling right now, and I need to scroll down. So the billing group here, again, if you'll have a pilot funds group, but there's probably no funds in that. So I have added everyone who gave the ID ahead of time to this Charge Workshop 2022 billing group. It's a billing group that I had set up and is being supported by NHLBI for this workshop. So you can run analyses in that billing group during this workshop. We'll close it down sometime in the future and let you know when it'll be closed down. So if there's files you want there, you can migrate out. You can choose the location. So Seven Bridges allows compute and storage on either AWS or the Google Cloud platform. So that allows you to avoid those egress costs that I talked about. So if your data exists on Google Cloud, you can run your compute in cloud and avoid in Google Cloud and avoid egress. Same thing with AWS. I'm going to stick with AWS right now, the Amazon Web Services. There's a few other options here that we can get in. Can't seem to scroll down. There's a few other options here that we can get into later, such as spot instances and memoization. These are ways to also help you control costs, which if we want to talk about in detail later, I'm happy to. But when I create a project, it'll bring me to that. So this is your view on a new project that you've made. And to orient you again, there's a description over here. There's full markdown language where you can write in whatever you want there. A lot of people use that as notes for that project. You can write in dates and keep a track of the analysis that you're running. And then you can invite new members to that project. So if you're a PI, maybe you want to add members of your lab to that project.
Sorry. On the Zoom, which we're trying to record, there's an unfortunate looking box on the left-hand side. So I'm just going to stop the share and then start the share again and hope it looks better. Well, participants, you can see our screen. Hopefully that will look better. What? No? Well, it doesn't, there's no problem on our side. So guys on the Zoom or for our recording, I don't know what we can do. So we'll just go. We'll go for it.
Let's try once more. Yeah. For good fortune. Hey, luck. Still no? All right. We'll just keep going then. Sorry, folks on Zoom. So you can invite, and I mentioned collaborators. That's one of the part of the power of the cloud here is your ability to collaborate. I remember when I was doing de novo genome assembly, we had a colleague in Europe that had a whole bunch of sequencing data and had to mail a hard drive because that was the easiest way they could get it to our HPC was to mail it and we could re-upload it on our own. The cloud allows you to avoid that. So I can look up my colleague Dave, who's online right now. And I can select what kind of permissions he wants here, or I want him to have. So he can write, which would allow him to add, modify files and tools. He could copy the files without write permissions. He can execute tasks without being able to access the files. Or I could give him admin where he could delete the project, edit, add new people, etc. And when I hit invite, look at that, Dave is added to the project, and I still control his permission. So even after I've added him, I can say, whoa, wait a second. I don't want Dave to be able to do anything in this project. So I'm going to take all of these away, and then he can only look at the project. Sorry, Dave. Right now you have read permissions. So we have our project set up. What do we want to do? We want to add data, and we want to figure out how to analyze. Okay, so I mentioned two types of analysis and a couple different ways to add data. So Emily went over earlier, building a cohort and importing with picture. She mentioned the API project. That's right here under public projects. There's a picture API project. You can also, we have a data browser here. So this is not a cohort builder, but it is a way to quickly access hosted files. So you're not searching and building a cohort the same way you do in picture, but if you already have a study in mind that you want to access, you have a study in mind that you want to access, or if you don't have access yet because your ERA Commons ID is not active, you can flip down and access, there we go, open data. So we have some open data sets such as the 1,000 Genomes project, and I will show you that right now. Hide those tips. There we go. Hidden tips. right? So we can explore the data set in the data browser. And as I said, this allows you to filter files here. If I click on files, it tells me there's 6,648 files in the 1,000 genomes. Again, this is hosted data. So when I import it into my project, I'm not paying for that storage cost. It's hosted in the NHLBI cloud buckets. We can filter through based on things like file type, file name, chromosome. Chromosomes shows up here, analysis group, etc. So I'm just going to go through some filters to get that file number down. And if I refresh it, there's one file left. Perfect. So I'm just showing you right now how to import that into the project. So you can see here, sample ID, there's some metadata in this column about that sample for this 1,000 genome sample. And now I want to move that to my project. So I can hit copy to project and choose which project, that test project I just made. I can tag it whatever I want. So if you're moving in multiple studies really quickly, you can just tag them real quick and copy the selected files, and they are dropped into my project. Look at that, awesome. Okay, I want to analyze those now. So we have a public apps gallery. I mentioned there's 600 plus parameterized tools. So you can go to the public apps gallery up here, public gallery, apps. It'll bring you to this page, and then you can browse and search. I mentioned the Genesis tools. There was a question also about, I think, EPACs earlier. So if I look up the EPACs tools, we can see what benchmarking information we have for it. So there's a whole bunch here. We have the kinship matrix. There's 7 items that show up. If I look at annotate right now, and I think I need to expand this. There we go. We got to read all. So all the 7 bridges parameterized tools that we have, so that 680, have descriptions. They have details for the input and output information. If you're following along, you can scroll through, search for your own favorite tool. So this is not quite as detailed as the Genesis pipeline, but you see we do still have some benchmarking information at scale here. So 48 megabyte file, 293, 750 gigabyte file. And you can see what the costs were and what kind of instance, what kind of resources were required to run it. So you can see the 750 gigabytes that had 2,700 samples, cost $3 to run. And we can add that to our project, but before we do that, I want to show you how to edit. So let's copy it to our project. And we'll go to our project. And we can edit this if I can. Oops. There we go. So we can edit. So Ravi had mentioned that instead of coding CWL, if you're uncomfortable with that, there is a visual editor. And that's not where I wanted to go. I swear I had just clicked on this earlier. Yeah, it's right there. Edit. I don't know why I'm not opening the visual editor. Let's try a different tool. There we go. That's what I was looking for. There's a visual editor here, so you can drag and drop. Over on this side over here, you can see the inputs. You can see this is the Genesis null model tool. So there's a phenotype input, the GDS file input, number of variants, etc. You can see the outputs on this side. And so right here is the command that's being called, so calculate variant score. You can see the output from calculate variant score over here can then be strung into the next app. And so you can search over here for something you might be interested in. something upstream or downstream, so VCF to GDS. And I can drag and drop and import that, and then again, connect the inputs and the outputs. So this allows you to build those CWL workflows without actually having to write CWL. And it's freely movable, and there's parameterization. If we click over here, all of the flags that you would normally get, or almost all of them, depending on the tool, that you would normally get from writing command line are over here for you to select. Some of the tools like Plink, I think Plink has 250 flags or something along those lines. I don't think we have all 250. I think we have something like 100 for Plink right now. But if you find something on here that you need more of, or you need something deeper, you can contact our support. We have bioinformaticians that are willing to help. We want to support you in the process here. Do people have questions so far? Are people playing along with me, or? No? Silence. All right. Okay. We will keep rolling then. So interactive analysis. We have a few different interactive environments here. So once you're in a project, we have 3 interactive environments that you can play in. We have RStudio, we have Jupyter Labs, and we have Julia. Or sorry, not Julia, SAS Studio. Julia is in Jupyter Labs. So you can select those here. If you go to, again, in your project interactive analysis, you create the analysis, and then you can select which one you want. And there's some parameters here, again, to go back to cost controls and to talk about resource needs. You can select what kind of environment. So these are, again, pre-parameterized environments. You have controls once you're in to install whatever you need. But for instance, the RStudio environments have Bioconductor already installed in them. So we already have some packages, dplyr, for instance. We have TensorFlow environments for machine learning if you need that. So you can select what kind of environment you want, what version of R you want, etc. And then you can select the instance. And so we have lots of different instance types. And this is where you can select what kind of resources you need. So you mentioned that you might need a terabyte of RAM. We have high storage instances in here that have a terabyte of RAM that you could do interactive analysis on. They are, I think, i3 instances. And you can search here. 768 of RAM, that's getting close. But so you can select what you want, and when you select it, it'll give you what the cost is. So this is a large instance where you have a solid state drive with 7.6 terabytes, 32 CPUs. It's going to cost $2.49 an hour. We have much less expensive instances. I think the R5 here is 12 cents an hour for using. Okay, so unless if you need a lot of resources, I would recommend starting with a less intensive instance. You can change how much storage you need, and right here is another guardrail. So when we fire up this interactive analysis, it's going to be running, and when you close the window, it will continue running. So if you're running a pipeline and you want to close out, it'll keep running. But we put a guardrail in place called suspend time. You can change this and you can turn it off if you want, but we recommend keeping it on, and it's okay if you change it. So the suspend time means that when the instance is running, you can think about this as allocating a computer. You walk into a library and you say, I need X computer. I walk over to that one and I allocate that computer to me. The suspend time here, when it hits that wall, it'll shut down the instance. It saves whatever's there, right? So it exports that back, but it shuts it down. So if something is running, it will stop that analysis, but it also saves you money. What it prevents is from you closing out the window and it running infinitesimally in the background, because we don't want that, right? Because then you're just spending money. So you can turn that off with the switch. We recommend leaving it on. And then you can name it. So you can have as many of these as you want. You can have each pipeline in their own analysis window. So I can name it Tony's analysis, and we fire it up. And it does take a little bit of time. It's not instantaneous for launching. So I'm going to go back to the project here. I'm going to go back to the main screen. Okay, so I mentioned the second half here. We're going to move into the Genesis pipeline. I'm sure that's why people are here. They want to learn about association testing. Elisa had mentioned there is a project made, the Genesis tutorial, by our collaborators at the University of Washington, GAC, Genomics Analysis Center. We can go to that. So public projects here, you can see there's a bunch of them. The public project space is where people have published collections of workflows, or the Seven Bridges team have assembled collections of workflows with tutorial information, some sample data, so that you can hop in and use those as a collection, right? So the Genesis tutorial is one example. We have some collaborators at Harvard Med that put together a machine learning chest imaging pipeline. We have freeze-8 variant calling. There's a whole bunch of others. The picture API is here. There's interoperability. So if you want to move data between other cloud ecosystems, and some others, like the Human Cell Atlas is also here as open data for easy access. We're going to focus on the Genesis tutorial. These projects exist and you can copy them and use them as you want. So the Genesis tutorial project here has a guide over on one side, as I mentioned, in Markdown language, linked to the GitHub, that walks you through what to do with this project and what's inside the project. And so if we want to use this, we can come up here. We can explore the project first, right? So we can see what files are here. We can view the files. You can see their subsets of the 1,000 genomes, their subsetted files. These are really small subsets of chromosome 1, so it's easy to run tutorials. All of the Genesis apps and the kinship testing are right here, pre-assembled. So it makes it easier for you to access and run. Any of the public projects also have completed tasks in it. So tasks are apps that have completed running, so the CWL tools. So right here, you can see what it costs to run those based on whatever data is here. And we can click in and explore that further. So you can see what file they ran. You can see this ran this subset chromosome 1. Someone was asking about selection for, and if there's filters to prevent like the wrong file types going in. And I'll show that in a second, but that's functionally what's here. So you see this, you say, that looks cool, I want to play with it. Up here at the I button, the information, you can copy that, and you can copy it to one of your own projects, or generate a new project here, copy a Genesis for charge, I give it a title, I assign it a billing group. So if you're following along right now, you should assign it to the charge workshop billing group. I'm going to choose to allow network access. That's up to you. If you're planning on doing, we're not going to do API calls now, but if you're planning on doing an API call, you'll need to be able to call out, right? But if you want everything to be restricted, no analysis can call outside the platform, then you can block network access. And we'll run copy. And what it'll do is it'll clone everything in that project to your own project. So you can edit it, you can do whatever you want with it, and it won't affect whatever that core project was. Then to flick back over to the R analysis tab. It's preparing the analysis environment right now. It should launch any second. Are there any questions as I'm going through right now? Are people playing along? No? Thoughts? Concerns? Do people need more coffee? We got a question, or a concern, I guess. Yep. So the button to copy is this little I up here. So it's right next to the title. And then there's a little, when you press I, there would be a little button that pops up that says copy. Cool. So if you've ever used RStudio, this is familiar, right? There's nothing surprising about this. You see the R terminal. If I open a notebook, it'll pop up top, just like normal. We have the global environment where you load in variables, your file repository down here. As I had mentioned, there is a bash terminal in all of these environments, and you have root privileges. So if you want to get in and go wild and build out your own tooling, and you're comfortable with code, go for it. So as I had said, right now, I'm going to close this tab out, and I'll show you there's a stop button here. I have the timer set to 30 minutes, which means my R environment here will continue running even if I close this out. So I can go back to the project that I had run that in. That was this test charge project. If I look over here at Data Cruncher, it says running. So this is Tony's analysis. That's what we were just looking at. It's easier to see if I go in here and go to Data Cruncher, which is our interactive environment. It'll say, oh, it's running, right? So you can always go back and check to see if it's running. What will happen is when it hits 30 minutes of running, it will automatically save and shut down. So I can stop it myself if I want, or if I let it run, it'll stop on its own. So again, guardrails so that you're not wasting money. So I'm going to stop that. It's going to confirm, do I want to stop it? Yes, I do. And it's going to save that environment. So if there's, again, this is, if you think about it, you walk into the library or you log into your HPC and you go to a node or you go to a computer in the library, that's what's going on here, is that when you launch one of these live interactive instances, it's sending you to a node in the cloud and saying, this is your computer in the cloud. And so when you exit out of it, it says, take your data with you. So it's exporting that data back to your main project. Okay, just like taking the USB drive out from a computer. And so it takes a little bit of time to transfer the data back. So that's saved, that's done. So we can go to copying the Genesis project can take a little bit of time. I think mine is still copying. It could be because more people are copying it. So if we give that a minute, are there, oh, mine is loaded. Are there other questions or people following along? Are we in a good place? All right, fantastic. We're at 4:07, we have plenty of time. All right, so Genesis project, again, we can explore what files are here, they're copied. All this project, so there's 54 files here, has everything that we need to go through that Genesis pipeline. So I'm only going to show a couple parts of it. I'm going to show part of the interactive analysis, which would be converting the VCF to the GDS. And I'll show harmonization. I'm sure a lot of people here are more familiar with harmonization than me, but I'll show that in the notebook as they have outlined. And then we'll switch over and we'll show the null model and single variant association running through the CWL tools. So we have our projects, or we have our files here that we can look through, see what we got. We have our apps here, so the CWL workflows. They're highlighted here because these are older versions. So that's, I accidentally hit back. These are older versions, so we can update these if we wanted. Come here and update, and it'll automatically say, hey, this is the version that you have. You're currently using version 23, but there are new updates available. And so you can click update. Versioning is important for reproducibility. So if we, again, click into our app here, up here, you can see which versions are available. And as I had mentioned with the other apps, there's lots of information here that you can walk through for your own, figure out what do I need to run this? What are the input files? What are the output files? So we're going to go back to the interactive analysis, as I had said, and we will run the VCF to GDS conversion, and we'll go over the phenotype harmonization. So go to interactive analysis in our Genesis project copy. We'll open it. Genesis tutorial is here, and we can fire that up. And we'll give it a minute or so. Are there other questions about things that I talked about earlier? I know there were a lot of questions and we kind of cut it off to move fast. We can go back and talk about other tools or whatnot. Does copying and deleting a project cost money? I think the short answer is no. If there are, so these files that are in this project are hosted files because they're the 1,000 genomes files. So copying the project does not cost you any money, and deleting it does not cost you any money. If you had a project, so you can copy your own projects too. And if you had a project that you uploaded data to and you made a copy of it, that data is linked. So you're not paying duplicate for that data. You're only paying for the original copy. So if you have 1 gigabyte that is in that original project and you make a second project and a third and a fourth, et cetera, you're still only paying for 1 gigabyte. I think that should answer your question. Do you have another question? Or is that your question? Ah, wait. Other questions? That's a good question. All right, this should launch in a second. So So this does take time. It's not instantaneous because again, right, the cloud, we're yelling to Google or AWS, hey, we need a computer. Yes. There's a question in the chat. To clarify, interactive instances have access to all our files in our projects. Would we be able to import existing Jupyter? Yes. So questions in the chat. Do interactive instances have access to all the files in the project? That is yes, it does. Would we be able to import existing Jupyter or R Markdown notebooks and use them in interactive analysis? Yes, you can. So if you have them on your HPC or your local machine, you can drag and drop to upload or use the command line or API and send those to the server and load them up in our studio just like normal. Or Jupyter Labs, whatever you want to use. Close the chat out again. All right, so you've seen this environment already. See, all of these projects or all these files over here are the files that are in the public project. There's an HTML here that we could have viewed earlier when we were looking at the files. We'll flip back. I click into the tutorial. Solutions are here. So you see everything is viewable here. Even the R Markdown should show up in text. In our R environment, I can load these. So I can just click on them to load them. So Genesis tutorial. And it loads up the notebook up top here. See the normal R windows down here. And so we can scroll through. And so this is a walkthrough tutorial. So as I said, I'm just going to walk through really quick the VCF to GDS conversion. Elisa had mentioned that GDS file is the important input into the Genesis pipeline. And most of the TopMed hosted genotype files, or all of them, are VCF files. So there are whole genomes as well, but the VCF files are present. So if you want to use Genesis, this is an important conversion step. So right here it says, convert a VCF to a GDS. You can walk through the rest of this on your own time. But if I come into the code here, it's annotated. See, I'm loading the required packages, and I can click through, and we'll run our conversion, just like that. And it exports, you can see over here, just your normal environment. And this is set up, again, as a tutorial, so it'll display in this code box here what the outputs are. We can explore the GDS file as well. So one of the key values to interactive analysis, if you use this locally, is the visualization, right? So we can explore files and be able to graph, look at summary statistics and similar information. And you want to be able to clean your data and understand the data a little better as we're doing. So we can look at the head of the sample, so the sample IDs that are present. We can flip through the variant IDs. How many variants are in the file? What do the columns look like? All of your standard data and data exploration. So all I'm doing is just walking through. Here's visualization. Here's the histogram. Here's our frequency of variance. And so this is really the value to the RStudio or the Jupyter Labs environment is visualization here. And we have in other areas of the platform, and I'm not going to demo them today, but our Shiny applications for interactive visualization as well.
And close it. That might be the source of the weird. Click on and then just close it. Let's see if that fixes
What's
something.
going on?
Oh. Weird little box on it.
Oh. Some Zoom troubles, but I think we're okay. So as I said, I'm just walking through the this VCF to GDS conversion, the exploration of the GDS. More of this can be done on your own time as you walk through this. There's good descriptions here. This was, again, built for the Summer Institute of Statistical Genetics at the University of Washington. So I'm going to scroll further down here to the phenotype harmonization. So a lot of the use cases that we have for the interactive environment are for harmonization because that's an iterative process, right? As you are looking at variables, and you need to match them. And as we'll see here, this is using the same use case that Emily went over earlier of height. So as we walk through the exercise, we can load dplyr, which you might be familiar with, is a common data exploratory library or toolkit. And we'll load in our studies. So we can see our sample IDs again and the information such as the height over here. You'll notice that these are 2 digits. Look carefully at the output. Does anything look suspicious? And if we look at some of our other studies, we'll see these are all capitalized, but some of our other studies were lowercase, right? And so you can't harmonize that, or you can't, when you go to compare that, if you're running a regression, it's going to come up with, capital sex and lowercase sex, and that's not useful for us, right? So we'll rename those. And then we can look at summary information after renaming summary statistics. This is important information as you're exploring your data. And so this is where we can compare. Remember, there were 2 digits for height on one of them, but we see 3 digits on the other 2 studies. So we're all scientists here. One is in metric, one is likely in English numbers, right? So we probably have inches and centimeters. So now we need to transform that data again. We look at our summary. I don't know if that transformation worked. Oh, yeah, 2.54. I multiplied a couple times. That was an accident. You can see that's now 1,000 numbers. That's why we always check our work, right? I saw this 409 centimeters. That didn't seem right to me. I'm going to leave it right now. But as we go through, we'll be able to merge all of these studies into the same data frame. And now we just have some really, really tall people in our data frame, over 1,000 centimeters. We can look at balancing. We have 190 females, 185 males. And we can plot. So again, visualization, it looks like study 3, we have a lot of really, really tall people, over 1,000 centimeters here. That strikes me as an outlier. If I was actually doing this for science, I would go back and double check my data and realize what I did wrong. We can plot the information based on sex. And then we can use regression models to compare our studies. So that's what I'm going to walk through with the interactive analysis. We are now at 420. I'm going to switch over to the CWL tools. We will finish on time, if not slightly early. So I'm going to stop this analysis here in the Genesis public project. That default is set to 300 minutes instead. So that is 5 hours that it'll keep running. If you don't want it running 5 hours and you are following along right now, I recommend you click stop in that top corner. It'll confirm that you want to stop, and I say, yes, I do. And it'll save the results. You'll see the exporting. You see up here the price controls. So I said I would give you insight into the pricing information. So right here, as part of it, it says you spent 8 cents on this analysis that you were just running. After we start up the CWL tools, I'll show you the billing group, since I know, again, there's a lot of concerns around price, and I'll show you how that breakdown, how you'll be able to track that. So I'm going to close this window. We have this project open over here, and we're going to go to the CWL tools now. So we're going to start by running the Genesis null model, and then we'll run the single variant test, which is down here. So you can see there's also a VCF to GDS converter here as well. So anything that is in that notebook that walks you through, we have CWL tools for. And the difference here is whether you are exploring the data and learning and testing for the interactive environment, whereas if you want to scale up, as I mentioned earlier, if you're interested in working on 50,000 genomes, you don't want to do that in the interactive environment. You want more power. You can go faster within the guidelines of whatever you're willing to pay. And so that's where these apps really come in handy. Or if you have perfected a pipeline and you don't want to run code by code, you can wrap it into a tool and run it easier. So let's start with this Genesis null model. Scroll over. we go. There's a run button over here. So if I hit run, it's going to open a new window and say, what do you want to run? So task inputs, there's a question earlier about wrong data going in, and this is where it comes in. This field cannot be empty. So anything that's parameterized that's in here, our bioinformaticians have put in the flags for what fields can be empty, so are optional, and what type of data is required. So if we look here, GDS file, this question mark, it'll tell us what do we need? Suggested file types, a GDS file, and what's the description to go with there. So I can click on select, and it automatically filters for GDS files here. So I can select the subset of chromosomes and save. Conditional variant file. Variants by gene. PCA. Phenotype. So we were using height just as we did before with Emily's example. So there's the null model, height phenotype. Family, I can choose what I want here. Yep. And outcome. So I need to name the output files or name what phenotype column. And when I'm ready, I can come up here and hit run. He'll say, your task is running. So it is queued right now. If I go back to that app screen and I scroll over, oh, sorry, tasks. It'll tell me that it's running. See, running in stage 0 of 2. And now we can check, we can monitor this live. So if I click into this again, and we can do view stats and logs. So if you're someone who really wants to know what's going on here with the batching and scattering, see, it says this is just started, but you would be able to see how many CPUs, it would visualize that for you, and what the cost is per step in that workflow. So where I'm going to go right now is back to previously run tasks. So if I go back to the copy of my project, you'll remember over on this side, all the public projects have executed tasks. And so if I find our, there should be a null model, there it is, null model. I can click into the completed task and I can look at the output files and also the stats around that run. So over here on the inputs, you can see what files were used for this matrix, what ran, all of the parameters that you use. So all of this is saved, again, important for reproducibility. All of our output files are right over here on the side in the output settings, where they went, what folder they're in. We can view these and we'll view them in a second, but I came to the screen because I wanted to see what happened last time I ran this. So if I click on view stats and logs, you can see this is what happened with this app. So it ran on a single instance here. Some of the instances are being initialized. And then it's running over here. You can see the null model has run over here. You see parameters over there, start time, end time, how long did it run. What you see there is instance is that 5.2 large is what kind of compute power was used for it. And so that is something you can also select in the options of where you want it to go. And then we can view task logs. This one does not have a log. So for the output files, after a task completes again, we can view all of the reports here. So here's an HTML report from the height file. So these are all of your standard outs from the null model, right? So phenotype distribution, all of your graphs are here. You can export this data back to your computer if you want, so that you can then make figures out of it, do whatever you want, or you can make figures in the cloud if you so choose. There's raw view. I don't think anyone in here wants to read HTML for a raw view, but if you want to, it's there. And other metadata associated with this file. So all the files that are here have lots of fields for metadata. If you upload your own files, you can put this metadata information in. Oh, I lost my scroll again. Right, so we can go back to our apps. So our task, we can run as many as we want. We failed because I put the wrong files in. So it tells us, hey, you spent 3 minutes here and 1 cent and it failed. So what you need to do then is go back and put the right files in, right? So there is a notice here. You can see completed tasks, you can see running tasks, and you can see failed tasks. We can now run, we'll now run the single variant association. So if you want to test this on your own, you can look at the Genesis null model. Here, it will tell you what files were previously used in this instance. So again, if you want to walk through these on your own, you can do that. Then we can go to the single variant. We can edit and rerun. So if you already ran a task, you can reload that. And rerun it. You could change the files. It'll re-import all of the files that you had there previously. And then you can go in and change them, right? So if you want to run this independently across many files, you can. Or, as I said, over here with, say, this GDS here, you can bulk upload. So you can batch files. So you can select your 50,000 or that folder and say, just run the same thing on all 50,000 files. Go to our tasks, and we'll see there's 8 stages here running for the single variant association. Does anyone have questions as we're going right now? We're at 4:30, so we still have a half hour left. All right, the last thing I was going to show is the billing group. So everyone is interested in tracking their costs and understanding their costs. I showed you where to find benchmarking information inside the apps. I showed you when it completes, there's a cost up at the top. I'm going to show you the billing groups now. So if I go up here to my username and go to the payments option, it'll give me the billing groups. So the charge workshop group, as I said, this is a separate test account. So I'm not an admin, so I can't view that billing information. I can view my pilot funds. If I log in with my other account, which I can do in a minute, I'll be able to see that information. But in my pilot funds here, I have spent $3 that I didn't have in the account somehow. 7bridges allowed me to do that. So you click in here and you can see what is remaining. So as I said, when you set up a user pays billing group, or when you get your $500 of pilot credits, that'll be right here is what's remaining. So if you don't spend anything of your credits yet, it'll say $500 remaining. Pilot funds, total charges. You can see a charge breakdown. And so this is a summary of analysis broken down by tasks. So that's the CWL tools and workflows, and by data cruncher analysis. So that's your interactive analysis sessions. And then storage charges. So active storage charges is the files that are either derived or are not hosted data, so data you uploaded. Downloaded, so that's your egress cost, and then storage deduction. So if we, clicked on something wrong again.
So may I make a comment?
Yes.
So Tony said that the live data won't incur costs. So imagine if you have a really massive file, and then you that's posted. So you're not paying for the storage costs of this really massive file. But then you bring that, you work on that file, save it to your workspace. Then if you have a problem of that file, it was not necessary for you to create a copy, but you had. It's sitting there in your workspace, put it or the change the version of the file. That's what the drive file ID. That it's a new version of the file. It's different from the one that's hosted. It's still really huge.
Yeah, I appreciate that. Yeah. Yeah, so I use that term derived data. So that's anything that you have worked on that is not from one of the hosted buckets, anything downstream of that, any file that's in your project. So if we go to the spending details for the billing group, here is where you can get a more detailed breakdown. So I mentioned sometimes PIs will make different billing groups for different grants, and maybe you have different projects undergoing the same grant. So you can see all of your projects here, and you can see the breakdown for all tasks and all analyses. So if I had run in test project, if I had run 50 different tools and workflows, they would all show up and the cost would be listed under tasks. And if I had run 10 different instances of Data Cruncher, they would all be listed under Data Cruncher with a summary up here. So you can very, very specifically, very acutely track the costs for your billing group. Go back to my project and see how that single variant association is going. It's on step 3 of 8. It's been running for 4 minutes, and we can track the logs right now. So you can see where it ran. It ran the SVG GDS renamer and defining segments here. It's now on the associate single R. Again, you can see what instance it's running on, how long it's been running. And from that information, you can get detailed cost breakdown, right? So we know we have an idea of how much that R 4.8 extra large instance might cost and how long we've been using it. Other questions?
Billing breakdown. How much visibility somebody has on the billing if they are not the administrator? Hard balance how much are not the administrator of that and
Gotcha. Yeah, so as just a member of the billing group, you would not be able to see that breakdown. Unless if Dave's online, he can correct me if I'm wrong, but so you can see I'm a member of this charge workshop group here, but I can't click into it to.
Hey, Tony, do you want me to make that account admin?
He's a participant.
you wouldn't see the overall summary, you would see the per job.
So you can see all of that in your projects.
Okay.
Yeah.
Well, I mean, that's
Other thoughts, questions? All right. Oh, there's a question in the chat. Do you want to be admin Tony on the billing group? Oh, yeah, Dave, you can make me admin Tony. So Dave has admin privileges on the billing group as well, so he can make me an admin. So you can see, we'll see how this changes when I become an admin. You can also contact support at any time to say you want to know what your daily storage costs are on any of your billing groups. Send Seven Bridges support an email. We're happy to provide that information. You want total costs for a billing group, et cetera. Our support team is always happy to help. Dave said refresh, and look at that. See, now I'm an admin. Now I can remove the billing group. I can leave the billing group if I want. And I can see all the breakdowns here. So current usage, data cruncher analysis, total analysis. I can look at, see if anyone else is doing work in here. There we go. So you can see all these projects is everyone in here who is currently running analyses, and we can see what their breakdown. So have some cruncher analysis going on here, Genesis tutorial project. And so I will be here all week, obviously, and this project is up and the billing groups are there. If you had provided your ERA Commons ID, you have been added to it. If not, I am around. You can just tag me and you can go in and play around and figure out how this works. You know, and it's NHLBI is supporting you playing around and figuring out how this works.
I will say that there is tomorrow morning between 8 and 8:50 on the schedule in the global room. It says BioData Catalyst drop-in session. I'm sure Tony and I will both be there.
I will be there.
Global room tomorrow morning. Anyone come in and try things out or ask questions or troubleshoot any.
We've been sitting for 3 hours and 36 minutes or more, so have to let people stretch their legs or take more questions.
We will stick around.
Yeah.
But that, this is basically the end of the material that we have.
Yeah, this is pretty much the end of the material that we have. So thank you so much.